Voice is not audible.
A very good morning to one and all. On behalf of MMTT-2021 team, this is Bhakti, welcoming you all in this 7-day international workshop, Molecular Modeling, Tools and Technique to Perform the Molecular Modeling and Computer-Aided Drug Design, organized by the Department of Medicinal Chemistry, Nagpur, Guwahati. With due respect, I would like to invite Dr. Krishna Ondela sir for further proceedings. Thank you.
A very good morning, one and all. Good morning, everyone who joined virtually and physically for this international workshop on tools and techniques to perform the molecular modeling and computer-aided drug design. And I formally welcome all of you, all the dignitaries who joined online and offline. I formally welcome all of you for this international workshop. And this is a big event organized by Nagpur Guwahati and Department of Medicinal Chemistry in the new year 2021. So we are hoping that everything goes well and it's going to be the fruitful for all the audience. They're going to be learn so many techniques in the 7 days continuous workshop. So on the same day, 11th January 1922, the insulin was first tested among the human beings. On the same day, we are starting this particular workshop on CAD and molecular modeling. I'm hoping that this workshop is going to be leads to the development of new molecules, not only in India, maybe other countries, the people who join from other countries. So I'm hoping sincerely that it is going to be lead to the development of many more molecules in the history. So with this note, now I would like to welcome all the dignitaries who joined physically and virtually to lighten the lamp. And who joined physically, I request Dr. Hemant Kumar Srivastava, Dr. Tamilvanan, Dr. Amit Alexander, and Dr. Sanjay Kumar Banerjee to join for lighting the lamp and formally inaugurate the session. And I request the dignitaries joined offline, our director, Dr. USN Murthy, and today's chief guest, Professor Iqbal Kanchan, and all other dignitaries who joined offline. I request all of you to join for the inaugural session by lighting the lamp. Thank you, one and all. Now I would like to request our director, Dr. USN Murthy, to deliver the welcome address.
The chief guest of the function, Professor Javed Iqbalji, and my colleagues, Dr. G. Narahar Shastri, Director of CSIR, Next Jorhat, and other dignitaries who are attending this divine event, international workshop on the tools and techniques to perform the molecular modeling and computer-aided drug design wholeheartedly to the wonderful campus of Naipur Gauhati, which is the first national pharma institute in Northeast region under Department of Pharmaceuticals. There are 25 eminent speakers excellently lined up in the sequence, spread over a period of 7 days. There are few speakers from abroad, from Israel, UK, France, Hong Kong, Saudi Arabia, and Canada. There is another eminent speaker from the industry, that is a Schrodinger, Dr. Mrs. Madhavi Shastri. And these are all the value addition and backbone of this wonderful conference. The aim of this conference is how to perform the molecular modeling techniques. Actually, why to perform and how to perform. From the basic drug design onwards up to the high-end applications in the drug discovery will be taught both theoretically as well as hands-on session by the eminent speakers. And before going to this workshop, let me tell something about this Holy Institute Naipur Gauhati. This was the among 7 Naipurs. This is established. It's the 5th one actually in this, and it is established in 2008. And it was inaugurated. The foundation stone was laid down by late Ananthakumarji, and since then it is coming up very aggressively with the flying colors. I am very happy to share with the 5 faculty and 5 departments who have secured 11th rank in the recent 2020 NIRF ranking. And we have got the national, almost 6 national centers funded by DST, DBT, donor, and various organizations. This is the institute first one in the Northeast region to have the BioNest Incubation Center, and under which more than half dozen incubators are already taken place, and they are doing wonderful service to come out with a well-defined product. And the aim of the conference is whatever the technology, what deliberations are there, that at least they should be useful to the youngsters and young faculty, how best they can take it, the beautiful knowledge of the 7 days in their future research. And this is the, especially in the area of drug design. So this, with these few words, once again, I welcome all of you to this wonderful session. Thank you very much. And over to Dr. Krishna.
Thank you, sir. Thank you for your kind words. So now I would like to request Dr. USN Murthy to formally introduce our today's chief guest, Professor Javed Iqbal.
Dear audience, it's a wonderful session today because it is well decorated by our chief guest, Professor Javed Iqbal ji, who doesn't require any kind of an introduction. And I can say who is a towering personality in the area of chemical sciences. Professor Javed ji obtained his PhD from Delhi University in 1977, followed by postdoc in most prestigious universities, Cambridge and Oxford, UK, during 78 and 83. When you come to his breadth of experience, it is almost 3.5 decades. As already, he is in the Ranbaxy laboratories in 78, postdoctoral fellow from Cambridge University 78 and 79, SCRC postdoc fellow from Cambridge University 80 to 82, research fellow from Oxford University 82 to 83, teaching fellow from University of Montpellier, France 83 to 84. He was a lecturer and beautiful orator, and he is from Delhi University in 1984. He was assistant professor in the IIT Kanpur from 85 to 91, followed by professor in 91 to 99. He was taken up a position as a senior vice president from Dr. Reddy Research Foundation in 1999 to 2001. He was a director also in the CSIR Regional Research Laboratory Trivandrum from 2001 to 2002. He was heading 140 researchers at drug discovery research of both Dr. Reddy laboratories in Hyderabad and also Atlanta, US. He was a director of Dr. Reddy Research Institute of Life Sciences, Hyderabad from 2007 to 2014. Currently, he was a founder and chairman at Cosmic Discovery Private Limited, Hyderabad. It is our fortunate thing to have you, sir, on the board. And with this brief introduction, I request you to give your inaugural address. Thank you. Jai Hind. Namaskar.
Thank you, Dr. Murthy. It is indeed a pleasure and an honor to be chief guest of such a very, very exciting conference that I see with a lot of people who are going to speak next 7 days. Thank you also, Dr. Hemant Srivastava, for helping me out. I must apologize for this delay as there was some problem from my end in joining the MS team. In fact, one of the recent advances in drug discovery, drug design has been the contribution from the molecular modeling, followed by many of the new tools that have come up in the last 20 odd years. I think this conference clearly, excuse me, is a very good effort towards highlighting the modern developments by calling some of the most exciting speakers. And I must compliment NIPER Guwahati about this institute. I've been hearing a lot ever since Dr. Murthy became the director. It's always been in the news about doing so many things, and I must compliment Dr. Murthy and his colleagues and students of NIPER Guwahati to really keep up, because NIPERs are going to be the future in terms of providing leads in the pharmaceutical industry in our country. And I think it's a very, very important step by the government to set up such NIPERs across the country. I know that you have some very eminent speakers, some stalwarts like Narahari Shastri and Bharatam and many others. I'm sure you will benefit from their talk. I thought I'll just in next 2 to 3 minutes, I will just show you something as to what I think about about this whole area, which I actually started many years ago. In Dr. Reddy's, and I can tell you that we were the first in Dr. Reddy's in this country who had set up an independent drug discovery group in Dr. Reddy's. Now, let me just in next 2 to 3 minutes tell you as to what I think. I'm not an expert in this area, but I think I just thought that I should share a few slides. So what is the future of drug design in the era of artificial intelligence? Now, one of the very outstanding Nobel Prize that was awarded in 2013 was on biomolecular simulation in drug screening and design. This was awarded jointly to Martin Koplux, one of the stalwarts, Michael Levitt and Arie Warshall, for their pioneering contribution in the development of multi-scale models for complex biochemical systems, recognizing the important role that the theory and computational methods play as a direct and necessary complement to experiments. See, until recently, people always thought that the computational methods in chemistry or science is very different and independent of experiments. But this Nobel Prize clearly indicated that there is a lot of connectivity between theory and computational methods and the experiments that you carry out in your laboratory. Now, you know all that machine learning methods have been very useful in accelerating drug development. For example, in the process of drug discovery, machine intelligence methods have mostly been used in computational methods over the past few decades. With the booming era of big data, machine learning methods have developed into deep learning approaches, which are a more efficient way for drug designers to deal with important biological properties from large amount of compound databases. Similarly, if you look into deep learning approaches, one of the very interesting development, the convolutional neural network, the CNN, which is called, are most commonly used, which have made great progress in the computer vision community and has now been applied in drug design fields, including de novo drug molecule identification, protein engineering, and gene expression analysis. So the rapid development of deep learning concepts such as CNN, the molecular modeler's toolbox, has been equipped with potentially game-changing methods. The CNN really is now become a toolbox in the hand of molecular modelers, which is really indeed a game-changing method. Now, another deep learning approach is modern deep learning architecture will be useful for the coming age of big data analysis in pharmaceutical research, toxicity prediction, genome mining, and chemogenomic application. This is a very important, powerful approach now in pharmaceutical research. For example, a fundamental technology that can predict the resistance of free cancer cells to fluorinated pyrimidine anti-cancer drugs by deep learning from the morphological image data taken from the image. You see, you can take the morphological image data and you can tell whether the particular cell is resistant to these fluorinated pyrimidine derivatives, which are very important thing, because instead of screening hundreds of molecules, you could now resort to so-called, the imaging or morphological imaging data, which can tell you about the thing. So I thought I will just share with you some of these approaches that are there, but I'm sure your experts who are going to be very sort of useful to this conference with their years of experience and their knowledge in this area. But definitely it's a great initiative by NIPER Guwahati, which I feel will be very useful. So the design has a bright future in the era of artificial intelligence and machine learning. And all the best for our conference MMT-2021. Thank you very much.
Thank you very much, sir, for joining with us on this auspicious day and highlighting the importance of today's conference on molecular modeling and computer drug design. Now I request Dr. Hemant Kumar Srivastava to introduce the workshop to the audience and mention about what is going to be happening in the next 7 days.
Good morning, everyone. Respected Chief Guest, Professor Javed Iqbal sir, respected Director, Professor USN Murthy sir, International Advisory Board Member, Professor G.N. Sastry sir, Professor G.P.S. Raghava sir, Professor Bharatam sir, and several other eminent people who are going to join in the next 7 days. Faculty members, registered participants, colleagues and friends. Welcome to MMTT 2021 International Workshop. When we planned this workshop, we thought of doing it physically. We thought of inviting all these people in NIPER Guwahati, in our beautiful campus between two hills, as I explained by our director. But unfortunately, that could not happen, and then we are doing in a virtual mode. But I can assure you that it will not affect the quality of the workshop. We have made all the arrangements with all the speakers, and hands-on session will be given to 24 selected participants. But other participants need not worry about it, because they will be watching it on Microsoft Teams, on YouTube Live, and the recording will be available in YouTube for future use also. If you see the workshop schedule, you will see many eminent scientists, starting from Professor Sastry, from CSIR NIST, Professor Raghava, who is a world-renowned bioinformatics, and Professor P.V. Bharatam, a well-known medicinal chemist, and Professor Etienne Derat, Professor Sam Davis sir, Professor Hajmi Hirao, Professor Devesh Kumar. These people are expert in QMMM calculations. You will also see Professor Sandeep Pal, Dr. Madhvi Sastry, Dr. Deva Priya Kumar, Professor Jagannath Mandal, Dr. Nihar Ranjan Jeena, Dr. Chinmay Chaudhary. They are going to discuss about molecular dynamic simulations in drug discovery and related topics in drug discovery. Dr. Farhan Ahmed Pasha, Dr. Kunal Roy, Dr. Priyanka Banerjee, Dr. Priyadarshi Satpathy, and Dr. Manavalan Balachandran, Dr. Sandeep Kumar Dhanda, and Dr. Harinder Singh. They are all going to talk about machine learning techniques, artificial intelligence techniques, and several other advanced tools and techniques in molecular modeling and drug design, as rightly said by our chief guest, that artificial intelligence is the future of molecular modeling and drug designing. I am thankful to Schrodinger, to Gaussian, and also to Jastra for arranging the hands-on demo in a short notice, and all the participants are going to get so much benefit about it. I hope you all have installed the Microsoft Teams, and basically we are going to give a full package. we're not going to give only machine learning. We are not going to give only like QSAR. It will be a full package. It will be like computational chemistry, computational biology, bioinformatics, chemoinformatics, and several other tools and techniques. This will be used in the next 7 days. If you have any technical issues, you can always contact our system engineer, and you have email MMTT-2021 MMTT@gmail.com. You can always write me an email, or you can write on WhatsApp. We are going to cover several open access tools like Autodoc, GrowMax, NAMD, and some commercially available softwares also. So all these information, it will be given to you in the coming days. And this workshop will start with a basic understanding of Linux and VI and Python, which will be in the afternoon session after the first inaugural talk given by Professor G. Narahari Sastry. And students selected for hands-on session are supposed to take 5 minutes presentation. This is already given to you. So it will be on the 15th, it will be on the 16th January. So you all are supposed to give 5-minute presentation, and the best one will be given Best Presentation Award. And let's make this program a grand success. Thank you all for your time.
So once again, on behalf of Naipur Gawati, I thank everyone joined here for this inauguration function. And I'm sure the 7 days workshop is going to be fruitful, and you are going to learn many techniques in the area of medicinal chemistry. And with this, we are going to be close the inaugural session. And by sharp 11:30, you're going to be have the scientific sessions. Thank you, everyone. Yeah, so now all of us can go for high tea, and who joined virtually also you can go for tea, and then you can come back. Thank you.
So Chief Guest, sir, thank you so much for joining, and very sorry for the interruption, whatever happened.
No, it's okay. I think there was some problem from my end, and I must thank. I hope that I have been able to do, but sorry about the whole. I think there is some technical problem from my end as well.
We are very much in time, sir. Our this session is supposed to end at 11. We ended 15 minutes before, so we have 15 more minutes in our hand. Thank you so much, sir. And the first talk will be given by Dr. G.N. Sastry at 11:30. Till then, we are going for the tea. Thank you, sir, for joining.
Thank you.
Thank you, Javed ji. Thank you, Namaskar.
Thank you. Thank you.
Thank you, Director, sir, for joining and for your welcome address. Thank you so much, sir.
Thank you very much. It's our function. Okay. All the best.
Thank you, sir.
Hello. Hello, Dr. Hemant, or someone is there? So Hello, good morning, Hemant. You are muted.
Good morning, sir.
Good morning, Hemant. So we have a few more minutes, right? About 8 minutes. You are able to see the slides, right? Slides are shared.
Your voice is not coming.
Oh, voice is not coming.
I think I have to increase the volume. 1 minute, sir. Yes, sir. Yes, sir. It was from my end.
Hello.
Yes, sir. Now it is audible.
Now it is coming. Okay.
Yes,
Okay.
yes.
So we have about 8 more minutes. The slides are shared, right?
Slides are shared, sir. Yes, it is visible.
Okay. Okay. So I will wait for
Yes, sir.
7
So
more
we
minutes.
can we will start sharp at 11:30.
Sure, sure.
Thank you, sir. Good morning, everyone. Welcome to the introductory talk on the International Workshop on Tools and Techniques to Perform Molecular Modeling and Computer Drug Design. In the first talk, we have an eminent speaker, Professor G. Narahari Sastry, Director CSIR NIST, who is also my mentor, and I am very pleased to introduce him. He is a pioneer in the field of molecular modeling and computer drug design, and computational chemistry, computational biology, and cheminformatics. Dr. Sastry has obtained his early career in Khammam, Telangana, and obtained his BSc and MSc from Osmania University, Andhra Pradesh. He is PhD from University of Hyderabad. After a couple of postdoctoral studies, he started his independent career in 1997 at Pondicherry University. He moved to CSIR IICT in 2002, where he headed the Center for Molecular Modeling. He is a professor of ACSIR in chemistry and life sciences discipline. Dr. Sastri's research interests are theoretical and computational chemistry, computational biology, CADD, cheminformatics, and software development in the area of molecular modeling. He has made fundamental contribution in the area of non-covalent interactions and developed several important concepts in this area. His group is interested to apply the data science approaches and also developing indigenous software, molecular property diagnostic suite. Several of his computational predictions have been experimentally realized. Besides publishing independently, the group also has active collaboration with several experimentalists and strongly believe that theory-experiment interplay is indispensable for the progress of science. He has successfully guided 21 PhD students and published more than 275 research papers in high-reputed international journals. Currently, 12 members are doing PhD and postdoctoral studies in his group. These publications received over 9,000 citations with an H index of over 50. Dr. Sastry has got so many awards that includes J.C. Bose National Fellow. He was awarded with India's Nobel Prize, Santi Swarup Bhatnagar Prize in Chemical Sciences in 2011, and National Bioscience Award 2009, and Swarn Jayanti Fellowship, B.M. Birla Award, B.C. Deb Memorial Award, CSIR Medal, and so many other awards and so many other recognitions for doing his science. He is very successful. He is a leader in this area, and let's welcome Professor G. Narahari Sastry for the talk. Thank you all.
A very good morning, and thank you, Dr. Hemant, for your very nice introduction, which I don't think that I fully deserve, and also for your invitation. And I would like to thank at the outset Dr. U.S.N. Murthy for giving me this opportunity and leading NIPER Guwahati from the front, and then involving people to work in various activities. And computer-aided drug design is one area which appears differently to different people. And we are so happy to have Professor Javed Iqbal, who had a very good blend of academic and industrial research in his career, which spanned almost over 4 decades. And in the next week, we have a very interesting line of lectures which will try to do some justification to what are the tools and techniques to perform molecular modeling and computer-aided drug design. The title of the talk that I gave is Nuts and Bolts of Computer-Aided Drug Design. While I am expected to go to the tools of computer-aided drug design, I would like to probably spend a little bit more time about the general challenges that computer-aided drug design throws and what are the tools that we are using to address these challenges in designing a drug molecule which will be effective against a particular disease, right? So let us have a historical perspective on this, where people have systematically looked at vaccines, serums about 200 years back, and also therapeutics from natural products mainly. And some understanding of how some of these natural products can be useful were beginning to get attention of so-called scientists, and the rational attempts towards getting a drug has started over 100 years before. Like, for example, we have a class of compounds like morphine and many others which were used as painkillers, because in human history we have health, and this health has been taken care by various measures which broadly categorized as health care. But when a person has an impairment of health which require medical assistance, they have medical care. Medical care is clearly associated with alleviating the pain, alleviating the inflammation, or any particular state which we call it as a disease. So an understanding has come that when somebody is having a disease, a particular small molecule or a herb or a natural product will alleviate the disease. And this particular understanding that we will have some molecules or medicines or aushad that we call will have a remedial therapy against a disease. These are called therapeutic agents. Then there are a large number of developments that have taken place in the last several decades, where people are trying to look at the gene. Human Genome Project has come, and then there is a omics, and there is a target-based therapeutics, and there is a molecular mechanism. Clinical indications of the targets were identified, and they are also looking at the phenotype-based studies. And people are trying to look at monoclonal antibodies and other biologics, where, as I list, the list is sort of endless, and a large number of developments have taken place in the few decades, where drug discovery has become really a very, very challenging subject. Now let's try to understand why a particular drug acts the way it acts like a drug. What is the mechanism of action? Or do we actually ask even a simple question that, do we need understanding of why a particular molecule acts like a drug? Because a large number of molecules were used as drugs and nobody knows their mechanism, either at the time of finding, or there are controversies, and the clear mechanism of a particular molecule and their molecular action is probed only in recent times. But historically, people were not so much bothered about finding what is the mechanism of action, because it's a very complicated procedure. For example, take a molecule and you orally administer it. You orally take it, and then it has to go to the gastrointestinal tract with varying pHs, which will go from very acidic to basic, and the molecule has to survive, and it has to survive the first pass mechanism, and it has to go to the bloodstream, and it has, because most of the time the mechanism of action is associated that a particular drug molecule interact with a receptor, and the receptor is more often than not lies inside a cell, and the small molecule should cross the membrane and then reach a target and reach only that target. Many times a particular molecule will be very efficient that this will do the job, but there are side effects because this particular small molecule will interact with other receptor which it should not interact. And there are things like human intestinal absorption and permeability is very important. And blood-brain barrier is also important, ADMET properties are important, and do we really require this understanding of the mechanism of action? It's too complicated. However, there are things like what is the relationship between a disease, drug, and do we have a relationship that a small molecule will bind to a receptor? And if the receptor structure is known, then can we develop a method based on the structure of the receptor? These are known as structure-based approaches. Or most of the time, we may not even know what is the mechanism of action, what is the actual receptor. But there are a large number of small molecules which will show varying levels of efficacy against a particular target. And these are called ligand-based approaches. So once you try to develop why a drug acts the way it acts, then you will have to see how do you decipher quantitatively this particular mechanism of action, or how do you even engage these particular data that is available for the benefit of designing newer drugs, more efficient drugs. What do you mean by more efficient? A particular drug, every drug will have some side effect or other. Is it possible to maintain the side effects optimally and increase the efficacy of a compound or increase its economic value? So can we make it cheaper, or can we make a drug long lasting, the dosages, and so on and so forth? So in all these aspects, knowledge-based approaches have played a very important role. For example, a particular molecule is biologically active. It shows some sort of a therapeutic potential. So most of the attempts are centered around that particular molecule and see how we can enhance the property of that particular molecule to make it better drug. So the approaches like Me Too molecules were very, very popular, and they are there in the drug industry, pharmaceutical industry. So what is the basis for this? The basis is similar molecules will have similar properties. Why don't you look at similar molecules and then see whether these molecules, for example, there may be a molecule which is very effective like a drug, but it might have some solubility problems. So is it possible to increase the solubility of a particular molecule? A particular molecule will be efficient, but it has some bioavailability problem. So is it possible to make some sort of a drug delivery system which will enhance the bioavailability, and so on and so forth. So there are different ways of improving the rational design of making a molecule into a drug based on the knowledge-based or empirical approaches. And other approaches which are more quantitative are to find out the relationship between the structure and activity. The structure of a molecule is a very complex thing to think about. It is a geometric structure, it's an electronic structure, or it has, maybe there are several other features of it. Is a structure of how is its electrostatic potential or hydrogen donating potential, the hydrophobic structure or hydrophilic structure, and which particular surface of which particular area of the surface will be electropositive or electronegative will also be very, very important to design molecules which will have a particular activity or a particular property. So attempts have been made to find out what is the relationship between the structure of a molecule and its activity. So SAR, and if you make it more rigorous mathematically, this is called QSAR, and a number of methods were developed based on a concept called lock and key model, where you will have a small molecule bind to a receptor, and this led to structure-based approaches, ligand-based drug design approaches. There is also an important concept like pharmacophore, or look at some features which will make it like a drug, and then a number of procedures are available to screen molecules in silico. This is called virtual screening. So I'm sure that in the next week, you will be exposed to a number of these techniques with a lot of theoretical rigor, methodological explanation, as well as demonstrating using the hands-on sessions. So there are several assumptions that one has. Some of the structures are similar, molecules have similar properties. Is it true? If so, what is similarity? Because it's a very, very difficult thing to talk about similarity, and I think when our group is trying to talk about the molecular property diagnostics or demo, and they will also be trying to tell you how to quantify similarity, and I'm sure that there are other people who are also trying to see the rigorous ways in which we can come up with matrices which will show how similar are two entities, whether it is molecules or something else, which are otherwise very difficult to subject to mathematics. And another assumption is a drug interacts with a receptor to elicit biological response. So this is true, but will a drug interact with only one receptor, or it can interact with multiple targets, or it can have different other properties which will be eliciting its biological response, and maybe interacting with the receptor is not so much important. There are things which are based on quantum mechanics, things which are based on physical chemistry, biophysics, physiology, and all these areas which appear to be very different. I put only a few areas because quantum mechanics is a rigorous way of evaluating the properties of molecules at microscopic level. So we need to understand what is microscopic and what is macroscopic, what is the property of a single molecule, or what is the property of a bulk matter. And quantum mechanics play a very important role, but quantum mechanics cannot be applied to large molecules. So one has to take also classical mechanics into consideration. But when you go from single molecule to a matter, which is absolutely important, one has to go to statistical mechanics and thermodynamics, where physical chemistry based principles, free energy, solvation, and all other thermodynamic properties play a very important role. And biophysics is another area, and physiology another area, and there's several other areas which will throw different challenges in drug discovery. And subjecting this to a very simple concept like this, like there is a receptor and there is a small molecule, it will bind with some non-covalent interactions, is a very, very simplified approach. So if you take an example from chemistry, molecular recognition is that you have a small molecule interact with a large molecule, whether it's a protein-protein interaction or protein ligand, enzyme, antibody, drug receptor, or whatever it is. But in this process, neither the small molecule is rigid nor the protein or the receptor is rigid. This recognition is not an event, it is a process involving two dynamically changing structures. And these structures will have completely different properties according to the environment. And temporally also they will be fluctuating and changing. And when different small molecules bind, they will have the ability to change the structure of receptor in different ways. So trying to trivialize that you have a receptor and it will have a shape, the lock and key type of a model, will be a bit too simplified. And as many of you have studied, that induced fit, which is a little different, is also one of the variants. And then people have to have the actual picture by finding out the three-dimensional structure of a receptor. It might be mostly it is a protein, and how it is undergoing the solvation and desolvation process. Because under physiological conditions, you all have to realize that the protein is solvated and the ligand is solvated. And after the process of the ligand binding to the protein ligand complex is solvated. So the free energy differences are very, very challenging to understand. Okay, so how do you find property of a molecule or a material or a matter? Let us go a little bit more fundamentally. What is hydrogen? People say that hydrogen is a gas, but that's not a very clear answer, because hydrogen is a gas at room temperature, but at some very, very low temperature, hydrogen will behave as a metal. And it can be even superconducting. And similarly, water will have completely different properties. Water property will be very different under different conditions. Water inside a cell is very different compared to outside a cell. If Schrödinger is giving a demonstration, you will know that there is a huge amount of discussion on a water map. And the phase diagram of water is one of the most complicated things to understand. Even now, people have a lot of discussion about how water capillary action and many properties of water are very poorly understood. If you try to take something like carbon, if I wanted to put in a very, very simple way, a diamond is also a carbon and a coal is also a carbon. But there's a huge difference between these two. So it all depends on how covalent and non-covalent bonds will form to make molecules very different. How non-covalent bonds will come together where different types of materials and matter will come from the same type of molecules. This is a very interesting subject in its own right, and there are ways of understanding the crystalline crystals, and their properties can be evaluated by a unit cell. But however, for amorphous molecules, for amorphous matter, it's a lot more challenging. And for something like protein structures and all, the challenges are very, very tough. How do you see the color of a molecule? Yeah, we can see physically with eyes or with some spectrometer. You will be able to find out what is the frequency, and from there you will be able to find out what is the absorption, at what frequency it gets absorbed. But how about the taste? The sweet, hot, salty, tangy, spicy are responses that these small molecules will have when they interact with the taste buds, which are also receptors, and which will give these types of taste to a particular organism called human being. But this taste can be very different to another organism. And similarly, if you look at the flavor, the smell will be different. Will be a particular flower will have a nice fragrance, and something else will have a different fragrance. But this is very specific to the interaction with these humans and a particular molecule. However, the same small molecule can have an entirely different taste, different smell with respect to some other organism. And even among the humans, there are different ways in which people will react to the same substance. Let's not get into it. So there are things like molecules. Somebody will say that this is a healthy one, and some molecules are poisonous, or something in between, toxic, or something is nutritional, something is unhealthy. And you can see that this molecule is hepatoprotective. or cardioprotective, or you can take any organ in the body, and then this will be good for bones, or this will be good for liver, eyes, or this will be good for endocrine system. And you can be more specific, there is an anti-cancer molecule, or antimicrobial, or antiviral, then anti-COVID, anti-TB. So there is a way of associating the property of the molecule with its structure. So I just wanted to tell you that drug discovery is triggered not only by the curiosity of the people, not only by understanding of how things are happening the way they are happening. It is actually the need of the hour. Drug discovery, whether you like it or not, one has to indulge in. And the only problem that we have is the type of challenges that we have are so big that no single discipline will be able to take care of this highly complex problem. Let's try to look at what is the world's leading causes of death. In 2019, people have realized that the heart attack is actually increasing from 6.8 to 8.9. And if you look at the stroke, it is also increasing from these are in millions, 5.5 million to 6.2 million in about 2 decades. And COPD is also increasing, but not by a big number, but things will change because of COVID. And lower respiratory infections are actually decreasing. And neonatal conditions are decreasing because of the healthy system that is there. And this trachea, bronchus, and lung cancers are increasing. And the neurodegenerative diseases are substantially increasing. And diarrheal diseases are decreasing because of probably the availability of clean drinking water and so on and so forth. But this whole scenario is what we have in a short period of 2 decades. However, one has to realize that the average lifespan is also continuously increasing. The average health of the people are also increasing. And I would like to bring to your attention that at the time of independence in India, the average lifespan is about 30 years. Now it is close to 70 years. So if you say that there is more number of neurodegenerative diseases which will onset after 50 years, it will be there definitely now, because the average lifespan was so low about 100 years before. And if you say that at that particular point of time this does not exist, it does not mean that people are more healthy. They are dying early. And similarly, if people say that diabetes is increasing, diabetes onset, especially the type 2 diabetes onset, will be after 50. So when more aged people are there in the population, the occurrence of the lifestyle diseases are also increasing. So there is a big need, or if you want to say in a very crude way, there's a lot of market in health and in drug discovery. However, we are in unprecedented times, and suddenly there are other things like malaria, malnutrition, homicide, Parkinson's. There are several diseases which are there, including poisoning, fire, alcohol, and drug abuse. But a particular situation called COVID has overtaken large number of factors, even about 7-8 months back. And this has become one of the largest causes for our deaths. So what happens? We all can have our own plans, but different things will emerge, and we should be prepared to understand how to really come up with better knowledge in dealing with the problems. For example, in COVID, we all have to look at how this host pathogen interactions can be controlled. Or specifically, if you are a computational chemist, you would like to know how this spike protein will interact with the host and how can we arrest it? And how do we do the immunomodulation in such a way that a particular host will prevent the pathogen from interacting? So this is a very, very important area. So knowledge is key. Knowledge is key in everything. It may be a drug, it may be a catalyst, or it may be a pesticide, or it may be a perfume, or it may be whatever. So if you want to design, you have to understand what is that molecule. So the question is, you have to know what molecule to synthesize, which particular molecule will have a particular property. Till 40 years before, people have a biggest challenge that synthesis of a molecule is one of the biggest challenge for organic chemists. But now, this how to synthesize, which was a big challenge about 30-40 years, was no more a big challenge because high capability was built in this particular area where people are able to synthesize just about every molecule that is conceived as a good molecule. So we have to identify a lead molecule which will be good against a particular disease, and then we will have to do the lead optimization, and it has to be validated. And the shift is from how to synthesize to what to synthesize from a medicinal chemistry point of view. So how do you know what to synthesize? The only way to know is you need to have a mechanism of finding the relationship between the structure and its biological activity, or in general structure and property. What type of a molecule will elicit a particular biological response? So the challenge is to hit at the right molecule, and in that way things are very, very demanding. The CAD market was expected to grow at a minimum of 12% or more. Growth in therapeutic areas, which was an analysis published about a couple of years back, that in oncological disorders, which is one of the highest, it's close to 50% growth is there, followed by neurological disorders and immunological disorders and infectious diseases. But this is the pre-pandemic prediction, and I think the infectious diseases might sort of comparable with oncological disorders if you want to do the analysis now. I just wanted to bring you to something which is completely different from drug discovery. You have to realize that there are complex problems that are there with the globe, that Earth systems are more intricately coupled. For example, El Nino in the South Pacific has been implicated to the outbreak of communicable diseases in faraway Bangladesh. Where is South Pacific and where is Bangladesh? And degradation of wetlands can be connected to bird flu and climate change, which helps to predict the outbreak of malaria epidemics in Africa. So we as a world are going into an area where the problems are all connected. It's important to generate the knowledge which will be able to solve problems which are common to the world. And it's not that it's all about a particular country. It's about all of us. It's like a Vasudhaika Kutumbakam that we have, that the world is one. Unless one has such a clear approach, you could see that a small mistake that has happened in a country like China led to a large number of other countries. Those infectious diseases is one thing, and also effluents are released into air or water. It can definitely contaminate globally. So the responsibility is very, very high to generate knowledge of how to tackle the problems. If you look at the R&D spending in pharmaceutical and biotechnological sector, it's growing very, very big. and it is flowing. The money is also flowing. But the question is in which direction? This is also very important. Just because there is a lot of money, it does not mean that all of it is being used in the right ways. There is still scope for people who will do intelligent ways in analyzing it and then come up with how they can use this particular area to make some big breakthroughs and contributions. For example, a conservative estimate from 2018 to 2024 is already a big hike. So this x-axis is the money that is there, the US dollar in business, and you have three shades of green, which has conservative, base case, and optimistic case. They are all very, very close. In all these cases, there is a definitive raise in the CAD services in 2024 and 2030, because this was an estimated 2018. And there are different types of therapeutic areas that are there. And driven by growing demand for effective therapeutics and increase in drug discovery efforts across various therapeutic areas, the market is expected to witness sustained growth in the future. So we have to have a quality manpower, quality thinking, quality infrastructure in the area of computer-aided drug design. This is a big area, and for a country like India, it's very important that people get trained, get motivated to work so that they will be very, very effectively contributing to the growth of this particular field, which is of immense practical applications. So the CAD service market, if you want to really look at the target identification, target validation, hit to lead, and lead identification are the areas. And lead optimization is one area. As you have seen, the top one, this play a very, very important role. And lead identification, hit to lead, and also target identification and target validation, all of them have sustained growth. So similarly, if you look at small molecules and biologics, small molecules will grow in a bigger way, and biologics are also growing in the same period from 2018 to 2030 is one estimate that was conducted by the CAD services market. They have published it, and it's a very, very effective study. And I feel that both academic and pharmaceutical companies will also be spending a lot of money in the services. The overall opportunity is anticipated to be well distributed across the various steps of drug discovery steps, types of molecules, and concerned sponsors. So if some of these students or young scientists are trying to get into this area, this is a very good area, and there's a lot of scope for innovation. And I would even say that only innovation will survive here. The conventional methods might be getting outdated much more in this area of CAD compared to other fields. For example, one approach like one disease, one target, and one drug. But this is probably not going to sustain. This is not sufficiently predictive. So there are different types of CAD service providers. I think it's repeated. And I would like to bring your attention to you that there is a large cluster of companies in North America, followed by Europe. Even including Asia Pacific, the number of companies are very, very low. And if you actually see the topmost column of this, we have put all Indian companies like GBK Bio, Syngene, Jubilant, Sai, all other things, Olivia. But still, the number of companies that are there in India and South Pacific and Republic of China, they are much less compared to Europe and North America. So there's a lot of scope for people in the next decade by 2030, I'm very, very sure that Asia will be a leader in this. So I don't want to really spend a lot of time on this drug discovery process. I just wanted to tell you that this target identification, target validation, assay development, hit identification, hit to lead optimization, preclinical, clinical. And in these areas, in every single area, computation, modeling, data science play very, very critical role, even in clinical trials, even in preclinical trials. However, in hit identification, hit to lead optimization and lead optimization, computation play an indispensable role, and there's a lot of scope. And I'm sure that in the next couple of days, in this next week, you will be coming across a large number of examples where computational biology, bioinformatics, molecular modeling, molecular dynamics, and other approaches play a very, very crucial role in the drug discovery process. As I said, not only in discovery, even in development process, they are playing a very important role. So if you want to really put things in a very, very simple way of drug discovery, there is a disease and you need to have a small molecule. Let's not get into biologics because this workshop is mostly about small molecules, if I understand correctly. And this area of small molecules was very highly explored, and the information that is available and the computational techniques that are developed and the algorithms that are known are much more robust in trying to design rationally. The small molecules are very, very high for small molecules. So how do I get the right molecule? Finding the Miss or Mr. Right for given a job. What is the job? The job is a particular molecule should become COVID-19 disease should disappear when somebody takes a small molecule. So the question is, what is that? Which molecule to synthesize? How do you know? How do you know, or how can you guess which molecule to synthesize? The simplest way is why these molecules behave the way they behave. Why a small molecule will become an anti-infective, an anti-COVID. So in other words, the mechanism of action need to be explored. How it is acting as an antiviral agent, right? Sometimes you get an answer from a computer software. The computer understands, but I also need to understand this particular reason why a small molecule acts the way it acts. That's not very easy. So we try to look at what are the nuts and bolts of these computer-aided drug design. Drug will act on a target. These targets are invariably protein targets, and they are involved in the metabolic pathways which are invariably linked to the genes associated with that, and a particular disease state is involved with these genes which are upregulated and downregulated. So a small molecule, when it interacts with a target, it will be effectively eliciting a biological response of therapeutic potential. So essentially, this is a relationship between the drug, target, pathway, gene, disease relationship, right? So what are the, if you look at the disease-specific drug development, which is the only way, because why do you develop a drug? Where is the need for developing a drug? Because there is a disease. So every drug that is developed is against a disease, right? So you need to understand what is the pathophysiology of the disease at a molecular level. What are the biomolecular targets involved in this disease? What are the genes involved? What are the organisms? What are the metabolic pathways? And more importantly, some of the molecules which are already known as drugs. Then one can ask a question that why do you develop a new drug? because these existing drugs will have some deficiencies. So it's extremely important to study what are the molecules which will have some potential against a particular disease, right? Then if you know that a particular small molecule is already a drug, but we need better drugs. So we need to know what type of a molecule to synthesize. It should have a similar scaffold. Probably that might not help. Maybe you will have to come up with different scaffolds. How do you come up with this? There are different methods. Like maybe some of us will explain to you in the next week about pharmacophore mapping or ligand hopping, or how do you get different types of small molecules which can be computationally designed. What will be the clinical implications? What are the side effects? And how this particular molecule is also interacting with already other drugs. How do genetic and epigenetic changes affect these things? So there are different tools, and I also would like to tell you that artificial intelligence is making a strong inroads in every walk of our life and every branch of science, engineering, and medicine. And drug discovery and development is not an exception. And in aggregating and synthesizing information, like collating the information available in various platforms, or understanding the disease mechanisms, or creating novel drug candidates, or doing whatever that is required, AI is playing a very, very important role, and it is essential that people should understand the role of AI and machine learning, which emerged as a lot more powerful tool compared to the bread and butter in computer drug design, that is biostatistics and statistics, which provided the basis for all the QSAR that we were doing, and also a number of virtual screening strategies that are there. But AI and machine learning has provided a new paradigm in drug discovery, not only in drug discovery, in every field, but also in drug discovery. The data, automation and artificial intelligence, and I would like people to this particular, although it appears to be very, very popular now, history of close to 100 years. So big data is a big term now, and I don't want to get into it, but I cannot really ignore it, because this is an important field where all of us really be exposed to it. You may like it or you may hate it, but you cannot avoid it. The big data science, artificial intelligence and machine learning, and it's especially utilize this in drug discovery. I wanted to now not some of the activities group that we, which will be available circles to pursue drug discovery. People have in drug discovery is that there are different softwares, and with varying experiences in different fields, he would like to develop an indigenous open source effectively provide a platform to bring which are necessary for drug discovery on a single. It will enable people to come. So this molecular property diagnostic purpose of strengthening computational drug discovery a little bit slow, but we are very steady in our and hopefully better than what already is there. Already we have this version 3-4 years because we started working on it. So the various modules that are there in MPDS is data analysis. We have also added data advanced modules are involving artificial predictive computing and multi-scale model. We are also involving very incorporated in molecular property diagnostic suite. This is independent and some of them are disease dependent. This particular chemical and also like available about a particular drug, what are the targets that are available, and what we can involve tools which are visualization involving statistical interaction, drug-drug interactions like this. Interactions. So platforms on this related to tuberculosis, which was published in years back in Journal of Biomedical Structures. Just wanted to tell you that this MPDS exposed to all of in the evening by our group, and we contribute to this particular software in your own ways. A fully open source software which will get into drug discovery without buying. Platform which is being hosted in CSI ICT, and we are also trying to host in a few other places, and I request people to ask any questions that they have during this 15th session. And we are very, very in biological space, and 130 million compounds which were curated to different classes and clusters, and incorporating different types of analysis, machine learning, deep learning, and data-driven approach. Different way. Is it possible to get which is 10 to the power of 60 or even amount of chemical space or the number of molecules that are to get 1 or 2 or 3 molecules which might be doing your job in a very effective way. So for that, there are a number of virtual screening, natural language processing and text mining can also be. What is most important is we have to knowledge that is available on a disease. proteins or the structures, and what are the small molecules that are there, information that is available, side effects, or what is the information has to be documented, and it has to be, one has to apply the state-of-the-art data science technique to get some valuable information. Is that one of the contributions from our group is that different classes and clusters, and this system given a very specific to molecule. This is very, very similar to a human being. We also would like to have an this particular effort is every information that is available on a molecule can Aadhaar card. So this particular done for 100 compounds that are there, and we are trying to expand it to, and more importantly, we are trying to data that is available on each of these molecules, so that this particular is useful, or we can use AI and deep learning from the existing data. Tell you that in 2019 we have added some modules based on AI, machine learning, and text mining, and hopefully MDS team actually started working in 2011. This particular manuscript is with all, and are able to publish a few papers in MPDS, and then now our team is, and this particular first paper has come as a cover page in Chemical Science, finished not only tuberculosis but diabetes disorders and MPDS for these are to be published, and hopefully in this year published. And the development of disease is a very interesting challenge. And completely disease, for example, or some other molecular dynamic or something related to chemical space or biological space, they are needed for any disease that we modules which are disease dependent and what are the drug candidates that are there. So we have divided these into disease dependent and disease robust disease independent MPDS. A small amount of effort, because they constitute close to 50 to 60 very, very tough and then we are now trying to make it more robust. And there are different challenges in post-COVID. There is a cost, what types of conflicts that we have, and you cannot wait for approval for a faster approval, and there are different cases require different levels of urgency. It's a completely different ballgame, which is very, very bad. Something like COVID. You cannot wait for 10 years. The world has been reacting very quickly. We should not forget what has happened in the administration of these drugs, unless they are clinical trial situations which are not very desirable. Time about we are working currently. I am working, and although people are from different parts of the country, region which has 8 states, 6 Assam, CSIR NEEST is the only CSIR division is advanced computation and data science division. Different expertise that is available in medicinal chemistry, natural products chemistry, synthetic chemistry, plants, geoscience, and we also have expertise in infectious. CSIR NEEST has a 2020 theme and 2021 theme is beautiful NEEST. The gate, when you enter, you can see on a wet day, there's 200 days out of 365. And we will be very happy to have you when you have time or when you visit your hut. Here we have biological sciences, engineering, science and technology division, and geoscience and technology division. And as I said, we have this. I am currently attached to, and also a new center for petroleum research. Working in areas which have a strong overlap with, need not be in new chemical entities, but in herbal formulations. I just wanted to, as it is one of different approaches that are available. In addition to new chemical entities, we have are very different. In Ayush mode, you don't have to go through the clinical trials, and phytopharmaceuticals is another way where there is a very good potential for India to contribute. And taxonomy and botany is very important because we will have to identify what is the different type of a plant and a taxa and a species, and morphotyping and genetic profiling and chemical and biological profiling is also important. And extraction, isolation, purification facilities are very important. And then we have state-of-the-art facilities in CSIR NEEST. And the biological evaluation is a continuous process, and we have a reasonable facility in our institute. In structural elucidation and chemical profiling, we have fantastic analytical facilities in our institute. So it's a good capacity that we have in the drug discovery area. And in health and health formulations, we have different verticals, and we are working a little bit closely on traditional knowledge and practices. And ethnotherapeutics in this area are very important, and we are also working in the data sciences for the well-being. And nurturing ethno-medicinal practices and traditional healers is another area, because especially in the Northeast, self-medication is done by close to 18% of people. And the next is people are going to traditional healers. Close to 40% of the people prefer to go to a traditional healer, whereas about 30 plus 14, a similar percentage of people will go to the modern medicine. Right. So there is a need for validating these traditional practices with modern science. So we are trying to see where are the gaps and what are the action plans, and the lack of unorganized platform or documents or database of traditional healers in India, in Northeast, is one gap that we have, and lack of documents and limited or complete lack of scientific and clinical validation, and also the strategies need to be put forward for sustainable conservation of ethnobotanical sources, and lack of school or training centers for traditional medical practices. This is an area where we are working on how to see that how do we develop the capacity building, and there are different action plans that are being planned. So in addition to that, CSIR NIST has a strong collaboration, including that with NIPER Guwahati and several other institutes, and we have been very effectively working in different programs like Aroma Mission, Immunomodulatory Nutraceutical Mission, Floriculture, Phytopharmaceutical Mission, and we are also trying to work a lot in the infectious diseases area, herbal product development, and data science driven research. And the ACDS, which have about 5-6 scientists and about 10 postdocs and PhD students, are involved in use of methods, algorithm development and applications for benchmarking protocols for virtual screening, docking, QSAR, network pharmacology, next generation sequencing, etc. And we are also having expertise in looking at quantum mechanics, molecular mechanics, statistical mechanics, molecular dynamics, and multi-scale modeling approaches. I just wanted to tell you that these are all very important for drug discovery. And it's very difficult for somebody to have exposure to every area, but it's essential to identify partners, because if somebody does not have expertise in this area, it becomes a limiting factor. And similarly, as I said, artificial intelligence, machine learning, IoT is another area, and harnessing Northeast biodiversity and traditional knowledge is something that we are working. And indigenous open source software for drug discovery, as I have already talked about, this MPDS is another area, and development of machine learning models and databases for chemoinformatics, toxicology prediction and drug discovery is also being pursued, and human resource development, and we also try to develop a portal for the health informatics. It's a very interesting statement by Nikola Tesla, whom I have a lot of regard as one of the finest thinkers and a very a great person in the area of science in last century. And he says that science is but a perversion of itself, unless it has as its ultimate goal is the betterment of humanity. So we all have to see what is the relevance, and during the COVID time, we are very happy that we are able to have this COVID testing lab, and we started with absolutely no experience in this area, and the lab was built within a month and it was operational, and we are able to do close to 38,000 tests so far. And we also have done this serological survey of all the employees of NEAST and many others, which is about 1,000 people, which provided a great base for us to understand these infectious diseases, and we continue to work in this particular area. Just because there is a problem, we should not really give up. We should come up with best way of tackling this problem. And another thing that we have done during this time is providing training. And when COVID struck, many people have difficulty in getting the summer research training. At that time, when we decided to give training, we thought of giving training to about 200 or people are like that. But when we announced 16,000 people have applied, and we are very happy that from June, July, August, we are able to give the summer research training program to whomever that has applied with the help of some sister laboratories. But a bulk of these people got training from CSIR NEAST, as we have been leading this, and with the support of Royal Society of Chemistry and Wellcome Trust DBT. So this particular training had students from everywhere. For example, this blue color is the highest. About 1,500 people came from UP and 1,600 people came from Maharashtra and close to 1,800 people came from Tamil Nadu. But you can see that every state has represented itself in the summer research training program, which was organized by us. And more importantly, we have 1 million visitors to our website. And during that, we also have conducted one-day workshop on artificial intelligence and machine learning, and also a webinar on chemistry and biology of natural products. And in addition to the summer research training program, we also have conducted many other things like quizzes, videos which will display the experimental procedures of a number of instruments, so on and so forth. And one of the star feature of this is very eminent people have given the lectures, people of the like of Sri N.R. Narayan Murthy, Padma Bhushan, the founder of Infosys, Dr. R.A. Mashirkar, the Prime Minister's Scientific Advisor, Professor Vijay Raghavan, DST Secretary, DBT Secretary, MOES Secretary, SCRB Secretary, many people have given, and this is led by our Honorable Director General, Dr. Shekhar Mande, and it was a very interesting experience. And at the same time, we also have conducted this Drug Discovery Hackathon 2020 training program from July to September, where we have given 3 different verticals. Vertical 1 is focused on theory and computational techniques, and vertical 2 is focused on the algorithms and method development, and vertical 3 is on hands-on sessions on open source and commercial software. And we are very happy that many people who are involved in this one-day MMTT workshop are also have played a very important role in the Drug Discovery Hackathon training program. Especially Dr. Hemant was there, he gave lectures, and Giri Pillai has looked at one of the vertical 3, and Dr. Nagamani and Hriday and Himakshi have looked at vertical 1 and vertical 2, and also they have overall looked at various other modules that are there, and all CSIR NEAST ACDS people are behind that, and the whole platform was created by 2 of my colleagues, Kirud Buragohan and Devabrata Das. And it's very interesting to see that once we create a platform, it provided all the necessary support, and the lectures are all available in the website. And if somebody is interested, I really recommend them to go through the videos that are available. Computational sciences, especially if you want to look at the atomistic details, there are limitations. There are limitations at fundamental level, there are limitations at practical level. I'm just trying to say that even at a fundamental level, there's a limitation. It's theoretically impossible to do things correctly. And in addition to that, there are a large number of practical limitations. What do you mean by theoretically impossible? Because if you look at any reality, it can only be measured to the limit of Heisenberg uncertainty principle. Any attempt to model the reality of an analog world with digital computers will have problems. We will not be able to have the digital computers depicting the world. So, whatever may be the accuracy that you are talking about will be depicting the epistemological reality but not ontological reality. There's a difference between these two. So our models, however honest, they will never be approximate. They will always be approximate. They will never be accurate. However, having said that, the innate motivation of any model has been to mimic the objective reality and closely as it can be perceived. Why I wanted to tell you this is, you try to understand the limitation of a model. Never ever come with an idea that molecular modeling is successful in some times or failure in some other times. Molecular modeling, computational approach will provide you a result based on a mathematical logic. Your result will be as good as your mathematical logic is. If your computational method is correct, if your algorithm is correct, it will be able to give you an answer according to the method. If a particular software or a particular algorithm does not yield a result which is correct, what it means is there is some inadequacy in the model. These inadequacies will be there because any realistic model will have to be approximate. So this is what one has to understand. I'm very happy that a number of people who are in the group are responsible. This is not all my group. This is just one picture that was taken. There is an advanced course in bioinformatics that was conducted. I would like again all the NIPER administration, Dr. S. Murthy and Hemant Srivastava and his team for taking event of organizing this workshop, and I hope all of you, and I will be very happy if you have any questions to answer. Wishing all of you the very best for 1 week of getting into the modeling and molecular model design. Thank you very much.
Thank you, sir, for very inspiring talk. Sir, we have a few questions which is asked by some participants. So one is asking, what is the limitation of computational drug discovery?
Your voice is breaking.
Sir, someone is asking, what is the limitation of computational drug discovery?
Limitation of
computational drug discovery. Sir, you are getting the voice now?
Unable to hear.
Sir, you are getting the voice now? Sir, you are getting the voice now?
Now I am able to.
Okay, sir. So one question is that what is the limitation of computational drug discovery?
What is the limitation? Limitation of
computational drug discovery.
What is the limitation of?
Computational drug discovery. Computational drug discovery.
Limitation.
Yes.
So, discovery is a very, very, if you fail approach, or if something, this is like a rocket science, because and computational drug discovery, I feel that it is even more complex designing a rocket. Hello.
Yes, sir. Yes, sir. We are getting your.
Yes, sir. We are.
Yeah. So, and reasons why a computer designing a drug will fail. The reason is that it may develop, or a drug may be defense. A drug might have problems of the solubility. It says the drug will be developed for a disease, or in some cases there will be another drug. Clinical trials 1 and 2, there may be another drug. Number of reasons a competition will not be commercially be beneficial. It is possible to understand the pathophysiology of if it is a drug against, and even in normal cases also, because there are things like efflux proteins that are there, a molecule will not be effective anymore after some time. So there, why the computational drug discovery view, and there are lots of limitations. But having said that, about drug discovery without computational approaches. Yeah, I think I have answered.
Yes, sir. Yes, sir.
Yes, sir. Yes, sir.
So another question is, can we call molecular recognition as molecular docking?
Molecular recognition.
So your voice is breaking. I assume that the question is, what is the
Molecular recognition
Recognition.
is molecular docking or not?
Molecular docking or not?
Recognition is a process. A molecule will go and then specifically receptor. They recognize each other. Where there is a specificity that is this particular, that is molecular recognition. However, molecular you try to develop a tool to bind with the receptor, both, and the other is a tool to or the small molecule bind is essentially anything, because you try to take a small molecule and you also look at the receptor, and then the dose of the small molecule is suited to, or will it fit or it will not fit. So it is There is a problem with the.
Okay, sir. So I think that there are a few more questions, but they are all very similar. So thank you for your talk, sir. It is very informative. And
Thank you very much. Yes.
yes, thank you, sir. Thank you very much, sir.
And the participants where the transmission. Okay, I thank you and very much for conducting this and giving me. And I look forward to listening to the lectures of us and also your lecture, Hemant. Wish you.
Yes, yes, sir. Yes, sir. Okay, sir. Thank you, sir. And thanks for being part and thanks for taking some time to join us. So as per the schedule, now we have lunch break and we will be meeting 2:15 p.m. sharp for the hands-on session where we are going to explain Linux, VI, and if time permits, some Python basics. So yeah, it will be a lunch break now. See you at 2:15. Thank you. turn on video and they can make an edit form, then you have to press I. I will make you in the insert mode. So now you are in the insert mode. You can see here it is insert. So for moving forward, you can press I. So if you press L, it will move the cursor forward in the same line. And if you press 6L, it will make the cursor 6 line forward in the same line. I mean 6 character forward in the same line. Same thing you can press, if you press H, it will take the cursor to the backward. And if you press 5H, it will take the cursor 5 character to backward. Now if you press W, it will take you 1 word forward. So earlier L was taking you 1 character forward, W will take you 1 word forward in the file. If you press B, it will take you backward, 1 word backward. Other than H will take you 1 character backward, B will take you 1 word backward. If you press J, it will take you down. You see, I am pressing J and the cursor is going down. If you keep pressing J, the cursor will keep going down. To press K, the cursor will move up. You can see that if I press K, the cursor is going up. If you want to go to the end of this line, you can press $. $. Will take you to the end of this line. If you want to come back to the beginning of the line, you can press cap. Cap is, if you press shift and 6, it will be called a cap. This will take you to the beginning of the line. Now, if you want to go to the end of the file, you can press capital G. Capital G will take you to end of the file. If you want to go to the beginning of the file, you can press double G. Double G will take you to the beginning. You see that my cursor is now in the line 1. And if you press capital G, the cursor will go to the end of the file. Suppose you want to copy line number 2 to 4 after line number 6. You can press colon 2, 4. You can press CO, CO is copy to line number 6 and enter. You can see that line number 2 to 4 is copied and pasted after line number 6. You can see this is line number 2, this is line number 3, this is line number 4. This content is pasted after line number 6 if you used the command 2, 4, that is line number 2 to 4, copy after line number 6. 2, 4, CO6 will do this job. If you want to undo this, you can press U, and whenever you press U, it will take you to the, it will cancel the edit whatever you made it recently. If you don't want to copy, you want to move lines, then you can do the same thing. 2, 4, you can press M and 6. This will make line number 2 to 4. This will move line number 2 to 4 after line number 6. You see that now this line number 2 to 4 is moved after line number 6. You can undo this. So this type of editing will make your life easy when you want to do deal with multiple files. Suppose you want to copy line number 1 to 10 to a new file. You can write like colon 1, 10, write, W means write, and you can give any file .txt or whatever you want. If you enter this, you can close this file. Close is colon Q, exclamation sign if you don't want to save the changes. Now you can see that line number 1 to 10 is copied to any file.txt. This was the information given in line number 1 to 10. This is copied to a new file. So new file is like this created. Now you can go back to the previous file. You can see that if you want to copy all this, you can do Control V. You can select this by putting the below arrow, down arrow. You can select wherever till whatever place you want to select. You can press Y, and then wherever you want to paste this, whatever was selected, you can press P, and whatever was copied, it will be pasted below the cursor.
So control what?
Control V, Y, and P. So, and now let's undo this so that it will not spoil the file. So I have undone whatever changes I made till now. So all these commands are really useful, and they will be helping you to do a few things. Like you want to change COMPND with something else. So you can press colon %s COMPND with MMTT 2021, and you press G, and if you press enter, you see that COMPD everywhere is replaced with MMTT 2021. Now you can undo this by pressing U, and if you do G and C, what it will do? It will ask you, it will select you COMPND, and it will ask you whether you want to replace it or not. If you press Y, it will be replaced. If you press N, it will not replace and move to the next. You press Y, it will be replaced. So GC means with confirmation. So wherever you will press Y, it will replace. Wherever you will press N, it will not replace. So this is a very useful command if you want to replace something with something. This is really, you suppose you want to replace remark, %s remark, replace with MMTT 2021, press G. See, wherever the remark was there, everywhere it is replaced with MMTT 2021. Now you can undo this by pressing U. Suppose you want to delete the current line where the cursor is. Suppose you want to delete this line. You can press D. It will delete that line. If you want to delete, if you don't want to delete the line, but all the characters written in this line, you press Shift D. Shift D will make it a blank line, and D will delete the whole line. So there is a difference between capital D and D. Capital D will delete the content of the line. D will delete the line. Okay. If you want to delete everything from the cursor till the end of the file, you can press D, capital G. It will delete everything. It will make this file completely blank. So let's undo whatever I made. Suppose you want to delete one word forward, or delete 3 characters. So if you want to delete 3 characters, you press 3x. You can see that 3 characters are deleted. You just undo this. TIT is 3 character. You press 3x. That 3 characters are deleted. Similarly, you can delete 3 words, that is DW. So it will delete 1 word. If you press 5DW, it will delete 5 words. So this degree is 1 word, and D is 1 character, E is another character, G is another character. So if you press X, it will delete 1 character. If you press W, it will delete the whole word. DW. Okay. So if you want to copy current line, suppose you want to copy this line, you can press Y. Y will be copy the whole line, and you can paste it by pressing P. So you can paste it multiple times. You keep pressing P, it will keep pasting whatever was copied. These are very useful commands which can be used by dealing multiple files. Suppose you want to extract some information. So let's close this file without saving, Q exclamation sign. We'll close this file without saving. If you press WQ, it will save the whatever edit you made and close this. If you press Q and exclamation sign, it will close the file without saving. Suppose there are several files which I downloaded from Protein Data Bank. You can see that so many files are there. If you open any file and you want to extract what is the resolution of this PDB file in multiple places, you can say graph this all the PDB file. What it will do? It will extract from, it will type the file name and it will type that line and it will tell what is the resolution. So for checking the resolution of each file, you need not open and see. You can simply grab it by using grab command. Grab command is really very useful because you can grab so many things. And for this, I think Anvesh will be telling you how to write a simple script. So I will not explain so much in grab, but I think it is really useful command, and you can do that. So just 1 second. So suppose you want to copy this file to another file. The command is cp file name to whatever file name you want. This will copy 1a4a.pdb to abc.pdb. If you don't want to copy, you want to move, the command is mv existing file name and new file name. It will move this file to abc.pdb. Now if you do ls, ls will show you that what files are there in the directory. If you type ls, you can see that this file does not exist now. It is moved to abc.pdb. Here is abc.pdb. Similarly, if you want to create a directory, suppose you want to create a directory of mkdir and wesh, Anvesh. If you do ls, you can see that Anvesh directory is created. Similarly, if you want to remove the directory, the command is rmdir. You can type this. This will remove the directory. But suppose you have a directory and you copy any file to that directory, and now if you use rmdir, it will say that it cannot remove. Then you have to use command rm -rf, and if you type this, it will delete the directory even when there are contents in the file. But if you type only rmdir, if contents are there in the directory, that directory will not be removed. So there is a small difference between these two. Suppose you want to copy multiple files to a directory. Let's make again this directory because it was removed, and it is always case sensitive. So if you are typing capital, it will understand. If you are typing small, it will understand. So case sensitive, it is always case sensitive. So now I made a directory called this, and you can make like this directory, different directories. You can see that this is one same spelling, but in caps and without cap. So these two directories are different. If you want to copy multiple files to any of this directory, you can copy this cp start.pdb and Anvesh. If you press this, all the PDB files will be copied to Unwaste directory. You can see that it exists here also. And if you go to this directory, which is CD to directory name, it will take you to the inside Unwaste. If you press ls, you can see that all the files exist here also. If you don't want to copy, you want to move these files, so you write again, like instead of cp, you write mv pdb to Unwaste, and then it will move all these files to this Unwaste directory. You can go to Unwaste directory and see that. You see that all files are moved here. Similarly, cd directory name, it will take you to the directory. If you want to come out from this directory, you have to press cd space ... will take you one directory back, .. will take you two directory back, and so on. So you can press this n number of times, but that should exist. So if you want to go to one directory back, you just press cd ... It will take you one directory back. Now let's copy all the files from this directory. You see, you can copy all the files from this directory, and if you press a dot, it will copy to the place where you are currently in. So you are currently in MMTT-2021 directory. You can copy all the contents from Unwaste directory, which ends with .pdb, to the current directory. If you press enter, you see that all the star.pdb files are copied from Unwaste directory to here. Similarly, you can move also. So, like this. Suppose you want to change this source with MMTT in multiple files. So you can do sed -i. You can type what you want to change. You can type MMTT 2021. You can do G prime and this file. So if you see this file now, wherever the source was written, it will be replaced with MMTT 2021. These things can be done in multiple files also. Now you see, if you do star.pdb, it will replace source with MMTT 2021 in each file ending with .pdb. Now you can see any file you can open. Wherever source was there, it will be replaced with MMTT 2021. It's a very useful command, because sometimes you need to do this while creating input file for running some calculation where you want to change one method to another method, and you have multiple files. You can use this command. Simply within one command, you can change to all the files which exist in that particular directory. Similarly, now I want to go back to the previous one. So -i prime s, because I don't want to spoil the PDB file. So let's go back. MMTT 2021 to source, G prime star.pdb, and then it will be done to the original. Now you see that MMTT 2021 is replaced with source. So these are some very basic commands which you need before you start doing Gaussian calculation and before you start doing Schrodinger calculation or whatever it is. But some basic GI commands and some how to create a file, how to move a file, how to copy a file, how to change, how to extract some information. In a very quick time, I try to give you some basic idea. Now you will be given a short tutorial of Python by Anvesh. So Anvesh is ready. I am just stop sharing my screen. Now he will share. But before he will share the screen, if you have any question, you can ask.
Hello, sir.
Yes.
So if we want to undo the last thing what we did, like we are replacing one word to another to any file, right? So how do we undo that?
So that's what I undo, no? Because you have replaced something with something. Now you reverse that. Earlier it was source was replaced by MMTT 2021. Now you replace MMTT 2021 by source. It will become the original.
Okay, got it.
Right, clear? So who is this? Can you introduce yourself?
Sir, Sanjana Rai Chaudhary.
So you introduce, you tell your institute name and what you are doing.
Sir, I am from Naipar Guwahati Medicinal Chemistry Department and I am Sanjana Rai Chaudhary.
Okay. Okay. Anyone else have any question?
Hello, sir.
Yes. Please introduce yourself.
This is Monica Verma from Medicinal Chemistry Department.
So you asked.
Sir, the same question which you were discussing now for replacing that. In that point, we cannot do undo like typing U.
No, that cannot be done. No, because you can be done only when you are open a file in BI. When you open a file in BI and you do some editing, then only you can press U. But if you are in the terminal, if you press U, it will be like this. No, how can you press U there? Okay. So for doing this, you have to reverse the process, whatever you have done. But if you open a file, in BI, whatever edit you did, you press U, it will be undone. You press Ctrl R, it will be redone.
Okay, sir.
Okay. So some people are suggesting that, writing that they are not able to access this and can only listen and don't ask question. So I will reply to their question through WhatsApp, and I will also reply through question by email. So don't worry about it. And then we will rectify this problem by evening and see that why some people are question directly. Anyway, so if any other question is there, maybe Pim, you are around? Pim, if you are around, you can unmute yourself. I think he is not able to hear me. Okay, no issue. So, Dr. Anvesh, please share the screen and then explain some basics behind Python. Thank you for that answer. Yeah.
Thank you so much, sir. I'm sharing. Okay, is my screen visible?
Yeah, now your screen is visible.
Okay. Good afternoon, all the participants for MMTT 2021. Thank you, Hemant sir, for my introduction and for organizing such a wonderful workshop. My name is Anvesh Pandey, and today I'll be taking a small hands-on session on usage of Python scripts in data mining. Like how do we use Python scripts to extract data from bulk data from files. So the contents for today's hands-on are what is meant by data mining, what we are trying to do. Python basics, we'll be introducing a few Python scripts, how to use Python, what are the basic command lines in Python, what are the basic modules that we'll be using, and things like that. Then we will understand the structure of a PDB file, at what kinds of information it has contained in it, and what are the possible information that we can extract out of that PDB file. And then we'll do a hands-on session and see how it's done. So data mining is the process of retrieving useful data from any dataset or any raw data. That is, if you are given a bulk file, bulk data, you want to take a few meaningful information out of it. Like suppose you have a PDB file, you want to take a resolution out of it. So as Sir demonstrated a few minutes ago, you can use grep commands also. But it is limited to say 1 files, 2 files, 10 files, 20 files. But suppose if you have 1000 files, 10,000 files, grep is not usually very successful method. So in that case, we can use PDB files. We can use Python scripting also. This can be done in batches, that is, and one by one also. So we can deal with one file at a time, and in batches also. This terminology is sometimes also known as knowledge-based discovery in data sciences. In this particular session, we'll be doing some retrieving some useful data from PDB files by starting from one by one and then by in batches. Python basics. It is a very powerful and effective programming language, and it is very easy to practice. Freely available for the Linux user, it is already there inside their system, and for the Windows, they can easily download it from the internet and they can install and use it. Start using it. There are a lot of tutorials and stuff available for practicing Python, starting from scratch to a very professional level. It is powerful because a lot of inbuilt modules are already available, which have a predefined set of functions, which we'll be seeing. A few of them we will be seeing in the upcoming session. For example, we have two programs are there, two Python scripts are there for extracting the sequence information and the source and the expression system, gene system of the system. So we'll be doing that. We'll see that. Okay, let's now start the structure of a PDB file and start the session. So is it visible? Can somebody confirm, is it visible?
No, the PowerPoint presentation is only visible.
The terminal is not visible?
No.
Is it visible now?
Your PPT is coming, and right hand side bottom, I think that is a terminal, right?
Can somebody confirm, is it visible?
No, please share it.
Yeah, now?
Yes, now it is visible.
Okay, thank you. So here I have 15 PDB files. I have mailed these details to all the participants also, and a few of the Python scripts. Now we will see, first of all, we will see a PDB file. So as sir already demonstrated, there are a lot of useful information in a PDB file. It is up to the user and it's a problem that the user is trying to identify the kind of information that we can retrieve out of a PDB file. So we have the title. We will be using a script to retrieve the title of a PDB file. We have the molecule ID. We have the organism scientific. We have the resolution. We have the sequence like SEQRES. Can you see the sequence? This is the sequence. We have the resolution. It's highlighted here. So we'll be extracting all these useful information from either one PDB file or more than one PDB file, depending upon the number of, depending on the type of work that we are going to perform. So let's see a Python script for extracting, first of all, a chain. So this is a Python script. This will extract the chain information out of all PDB files. We have the import module. It will import the system, that is, it will interact the bash, the kernel to the Python. It will call the Python module inside the kernel. Then we have system argument. System argument 1 means whatever module we have defined over 1, this will be called. Then we'll be opening the function and we'll have defined a variable which is known as chain A. Now the condition is applied while 1, that is system argument 1, and the chain is A. It will do what? It will split and read lines. It will read what? Whether if it is 0 to 6, if at line number 0 to 6, if SEQRES is present and Along with that, at line number 8 and line number 10, if there is one space and 1, it will capture line 11 and line 12. It will extract the data, the information from 11 to 12, and it will append into another variable known as chains. This chains will be transferred to chain A named variable, which we have already defined over here, and the data will be finally printed along with the function. That is the PDB file. This function will refer to the PDB file, and chain A, whether chain A is present in that file or not. We can increase this from chain A to chain B, C, as to n number of chains if present in that file. So let's see how it works and how do we do it. First of all, we will do it with one PDB file, and then we will do it with every PDB file. See, you can see the result flashed on our terminals that function and A. If we look at the Python script again, towards the end, we have given a print, that is the function along with the chain information. If we look at the output, we can see here we have the function, that is 3MWW, which is the name of the PDB file, along with the chain information, that whether chain A is present or not. Fortunately, this file had a chain A, so chain A is printed. Let's see if it works with all the PDB files or not. So as Sir demonstrated, if we have a lot of PDB files or log files or whatever with the extension, we mention it with star.extension name. See, we had 15 files, 15 PDB files along with the function, and their A chains is displayed on the terminal. Likewise, we can also see if there are B chains present or not. So we already have another script for it. Let's see. First, let's see the script. This time we have made a change in this script. Previously in this upper line, this Python 3 chain A.py star.pdb, what we did was we mentioned star means all the collections of the files ending with extension PDB. This we mentioned ourselves in the terminal itself. But this time in the Python script itself, we have imported another module named globe. What this globe does is it reads a particular extension or a particular path or a particular file type. Like I already mentioned, that Python is a very powerful language. It understands the demands of the users and the nature of the work, so it can be modified accordingly. So I did a small modification. Along with the system, I imported another module that is known as globe, and in that I have taken now, I'll be writing the output instead of displaying the output on the terminal as in the previous demonstration. This time I will be demonstrating the output in an external text file. This text file will be named as chain_ab.txt. This W stands for writing in that text file. What to write? It will read all the files ending with PDB. See star.pdb, which we have defined that globe over here. So this globe, what it does, it calls all the extension. If we have suppose log files, we can remove PDB by replacing PDB by log files. If we have any other file extension, we can do the same. This time we'll be mentioning along with chain A, we have chain B information also, whether there is chain B present in the list of PDB files or not, we can see. Now the similar pattern is there, seq res. First the program will look for in the line 0 to 6 for seq res, and along with this it will also look for space 1 at line number 8 and 10. If it finds so, it will extract the information, the data present in lines 11 and 12 and save it to a file names chain A. See, chain A is mentioned. Similarly, if the data has chain B, it will save it to another variable named chain B, and towards the end it will print. But since we want to display the information in another text file, which we have already mentioned in the third line of the script, chain_ab.txt, so this time instead of displaying the output on the terminal, the program will, what the program will do, it will take the output, it will write the output in a chain ab.txt file, and it will save the file. And later on, we can view the file in any text editor like Nano or VI. We'll be using VI here. So let's see and then see what happens.
Hello.
Yeah, I can hear you.
Yeah, hi sir, it's Suyash. So I have a question actually. So as you said that after SEQRS and line 8 to 10 means is it means if this SEQRS will be means after 10th or means after 12, 13, 14 or 15, what will happen? Means it will take
wait. Let me yeah, I got your question. Let me open a PDB file and see if 0 to 6 has SEQRS and how it's doing. Let me demonstrate you by opening a PDB file.
Okay sir.
Okay, let's open a PDB file 3mww /seqres. So Suyash, can you see the highlighted part?
Yes sir.
So SEQRES is already highlighted over here.
Yeah.
So it starts with 0. The counting starts with 0 and the last S is at the 7th place, but since the counting is starting from 0, so it will be counted at 6. Okay.
Yeah, so means all the PDB files follow the same pattern, so
Exactly.
we can use this same script can be used for means n number of PDB files, right?
Exactly. I'll be demonstrating the same script for one file and for all the 15 files. If somebody wants to use the same script for say 1000, 10,000 files, they can do it. The data has already been shared with all the participants.
Yeah.
So
thank you, sir.
Okay, no problem. So should I demonstrate? In AB, let's check with the one PDB file first of all. So as I mentioned in the script, we had created a file named chain AB and stored the information in that file. So let's see if there is any such file. See, we have a chain ab.txt file. So let's visualize it. See, what it did, it took all the PDB information from all the files, whether there is chain A or chain B or both. If suppose some of the PDB file had no chain A, that would have been blank, or if some PDB file had no chain B, that part would have been blank. But since all the PDBs had both the chains, we can see that. Again, let me share the script, the same script. We imported sys to incorporate the entire Python system. We imported glob to read a particular text file, a particular type of file, be it PDB file, be it log file, be it a text file, be it any extension. We created a file named chain ab.txt and wrote the appended information. We wrote what? We wrote the appended information. And how did we append the information? This is mentioned in this line, in the conditional clauses statements. All the appended, how did we append the information? It is mentioned in this these 4 lines. Later we printed that opened file chain ab.txt is opened and is to be written what? This the information which is to be printed, and finally that file is to be closed. So again we can, one more thing, let's see the chain A file. This has a line called system argument 1. We'll be using this and see how it works with n number of files and one files, as the question asked by Suyash. So we have only one file. Suppose if we take another file 3igv, See, we have two information now. So likewise, we can extract information, be it any kind of information. Now let's move on to extracting the title of the file. So let's look at the structure of the title file first of all. Similar structure is there. This time, instead of appending the chain information, we have extracted and appended the title information and saved it in a variable named dt. And later on, we have printed that dt and we have opened a file named, say, let's change the name of this file from info to title.txt. Now let's see if there is title.txt file. Last file, the last file you can see, it is title.txt. Now let's visualize it. This is the title of the PDB file as mentioned in the PDB file. Whatever be the title mentioned in that PDB file, that will be displayed either on the terminal or on the file which is being called and written. So likewise, we can demonstrate with all chains. We have a script named all chains. See, this time, suppose our PDB file has Z number of chains in it. So what are we doing is we are we again did the same thing, called the system, imported the globe. Globe is to read a particular extension. We defined our variables. Since we are assuming our files to have Z number of chain information in it, we have defined a Z number of variables. Suppose it does not have that much, it will be printed blank. Let's see how this script works, and then we'll be discussing about the working of this script first. See, so we have already seen in the previous that all the files had only two chains. Z chains were missing, but we deliberately created a script in order to find out whether by mistake we could not find any C, D, E, F or M, N, whatever chain name is there, and to look as if we by mistake mishandled a file and lost a chain information. So that way we can see whether we can verify whether what we are assuming is correct or not. So what is the difference between the two chained file and Z chained file? There is a simple difference. Chain ab.py. So here what we did was we assumed the files to contain only two chain information, and we created a program to extract information and print. Let's not print this on the on a file, but rather print the information on a terminal. So what we did was we put a hash. Okay, putting a hash before any line makes it a comment, and it's not it doesn't becomes the part of the program in that case. That is, when the program is executed and compiled, it doesn't count as to be the part of the script. It counts as a statement. Like I can write a statement over here that this is a Python script for information. Anything. We can write anything over here. This will not be the part of the command. Let's see if it works or not. See, so what is the difference between all chain.py and chain ab.py? We anticipated in all chain.py that probably all our PDB files had more than two chains. like A and B, it could have N, M, L, Z, X, A, anything. And possibly we would have missed that extracting the information by looking at the structure of the PDB file. So we scripted it and assuming that all the files have all the chain information. So whatever file has whatever chain information, it will be extracted and whether displayed upon the terminal or stored in another file, depending upon the nature of the work. Now let's have a look at some other scripts also. Now we have a ph .py. This script, what it does, it brings out the ph information from all the PDB files. So what are we doing is, from line 0 to 6, it will be searching for remark, and line 12 to 14, it will be looking for ph. And then later on, it will be extracting the information that is present between line number 45 to 48 and storing that information into a variable known as ph1 and finally printing that over the terminal itself. See, here we did not include the globe module. We did not create a file which is to be written and the data is to be stored. So let's see how this file works. This time we have to tell it as to what file we are trying to operate. PD5M. Okay. Let's see. PH1 is 45 to 48 append PH1. I think we have not defined this variable and therefore it is not taken. Okay. So what to do in case we find an error? The easy way is to look at a previously done file and to look at the patterns and check if what is the error. So we will be doing the same thing here also. We will look at a working information file and we will be seeing as to what is the nature of this file and how this file worked and that the previous file didn't work. Let's see. So what we did in this file, we took out the resolution information as well as the pH information, and this file fortunately it worked. Now let's see what is the information drawn from this resolution and ph.txt as mentioned in the third line, that it created a file named resolution_ph.txt. It has to write in it. What it has to write? It has to study the all PDB extension files and look for similar patterns like 0 to 6 for remark, 12 to 14 for ph, 45 to 48 for ph1. Similarly, for pH information, for again remark from 0 to 6, 11 to 22 for resolution. Mind here that the resolution ends with a dot. We have included the dot also as to make the information better. Then extracting the information between line number 26 to 30, storing that into the variables named res, and then printing out in the opened file named res_ph.txt, and finally closing that and saving that file. Now let's see how this works. See, it has the first column represents all the PDB files This column, the second column represents the pH, but this is 7.4, 7.6, 4.7, 5.4, and the last column represents the resolution of the PDB file. So that way we have extracted the pH as well as the resolution. Now let's try to see another gene information. So what it's doing is it's taking the gene which is from source to gene, and suppose, see, there is a column also. Let's look at a PDB file and see if this structure is present or not. 0 to 6 is source. If somebody want, they can write this. 11 to 16 is gene, along with a colon, and we have to extract from 17 to 57. First of all, let's execute this program. See, okay, this is a very nice information for us now. So in all the previous scripts so far we have seen all the information was present in all the files, but fortunately in this script what it does was we have last two files have this gene information and the remaining files doesn't have this information. So as I already mentioned that whatever information will be present in whatever file, only that will be displayed. If some file doesn't have that information, that will be remain blank. So we can verify it with the chain also by executing the all chain.py file. See, this had only, all the files had only chain A and chain B, but not C to Z. All the files doesn't had any gene information in that very particular extracted line 17 to 57. They did not have any gene information. Only these two files, the last two files had the gene information, so it is displayed for these two files only, and those files which did not have any such information in the previous mentioned lines, no information was displayed for them. So likewise we can incorporate various information by any PDB file and we can see if it works well or not. Now let's look at the residue and title also. We have a few minutes left. BI residue. So what this script is doing this script is simply extracting the residue information appended between line number 21 to 57. It will extract, it will append to variable named residue1, and it will finally print it. Since we have not used the opening the file and writing the information in that very named file, and again towards the end we save the file as in mentioned in the previous scripts, so it will display the output on the terminal itself. Let's see how this works. See, okay, another very nice information for all of us. See, let's check the file again. Between line number 21 to line number 57, the residue information was present. Since all the files have different residue information, we have the residue information for all of them as mentioned between those mentioned lines 21 to 57. Suppose for this file 3BR9, the residue was RNA directed RNA polymerase, but for 3H2L this information was NS5B polymerase. For another file it is something other information. So whatever will be written in that very mentioned line of the file, that part will be extracted and the remaining part will be omitted. This is how we can use the Python scripting in data mining. This process is known as data mining. That is, we are mining data. What miners do, they dig the earth, they bring metal, they bring the metals and sands and all that, and then they filter out the iron ore, the other sulfur ore, copper ore. So likewise, what we are doing is we are digging PDB files or log files or whatever extension files for relevant information. Suppose if we are if we have downloaded 1000 PDB files, but we have to dock only to those PDB files which have the resolution information less than 1 angstrom. So we can easily filter out of 1000 files what files have the resolution less than 1 angstrom or between 1 to 2 angstrom, as to how fine their crystal structure is. The finer the crystal structure, the better the docked pose results will be there. So all these things are linked in this manner. If somebody has something to ask, we can.
Hello, sir.
Yeah.
So in one file, we are typing the system argument.
Yeah.
And one file we are typing globe. So what is the difference?
Yeah, very good question. So let's first of all see those two kinds of. What is your name, please? Can you tell your name and affiliation, please?
So my name?
Yeah.
Sir, Sanjana Roy Chaudhary from Medchem department, Naipar, Guwahati.
Okay. So as Sanjana asked, let's first of all check the two kinds of the file, and then we'll see what are the differences between those two files. So Sanjana asked is that in some files we have mentioned this system argument as 1, whereas in some files we have imported a module named globe and we have read that globe. So what it does was, first of all, we have imported sys. Sys means our system is to interact with Python, and whatever interaction is to be done, 1 means the argument 1. 1 stands for to read the very first argument of the given line, command line. So very first argument of the command line is importing the system. Okay. Whereas in case of here, what we did was we did simply included the module named globe. Globe, as I already mentioned, it reads extension, it reads path, it reads a definite file, it reads a definite extension. It could be PDB, it could be log, it could be text, it could be any extension depending upon the user. So this is the preciser way of doing things, especially when we are handling a bulk data. In this script only we can see that a bulk information is extracted from either one file or n number of files. So in that way we can shuffle between system argument as to be 1 or system argument as omitting the system argument and putting globe and writing the star.pdb or star.log or star.txt depending. So in the very beginning of the session I mentioned that Python is a very powerful language, and all the power that Python gets is because of its modules. We called a module named globe, and that globe omitted us to write system argument 1. We just have to call the file extension. Sanjana, did you get the doubt?
Yes, sir. Like for globe, it is more specific than a whole
exactly.
system.
Exactly. Exactly. Anybody else with some doubt? So we have provided all these files and the scripts to all the participants. They can practice it, and obviously, as sir mentioned, they can come up with doubts. They can mail, they can WhatsApp us, and we'll be of course getting back with all the clarifications needed. Suyesh, are you there? Is there something that?
So thank you, Anvesh. Thank you so much for your nice session. It was very informative.
Thank you so much.
And all the hands-on participants are supposed to interact and talk in between, and they should also, while talking, they turn on their video so that it will be a real hands-on session. Now we are going for a 15 minutes break, and immediately after that, Gaussian tutorial will be given by Dr. Charitra Gaur. So see you after 15 minutes.
Thank you, sir. Thank you so much.
Good afternoon. So, welcome to the afternoon session of MMTT 2021 International Workshop. This is the first demo on day 1, which will be given by Dr. Charitra Gaur, who is working in Scoop Scientific in India, and he has technical expertise in computational chemistry softwares, and he has given such demo in various institutes, including IIT Patna, IIS Jaipur, Central University Jammu, Delhi University, Pune University, Manipal University, and several other scientific and research organizations. He has expertise in Gaussian, and today he is going to explain a few advanced topics of Gaussian. He has already completed some calculations, and he will tell you how to make the input file, how to run the calculation, and when your calculation is over, how to extract the information from the output file for your purpose. Let's welcome Dr. Charitra Gaur, and over to him. Thank you.
Thank you very much, sir. I hope I am audible to all the participants, right?
Yes, sir.
Okay, perfect. So let me just share the screen. So I hope the screen is also visible, right?
Yes, sir.
Okay, perfect. Okay, so first of all, thank you very much to Dr. Hemant and his team to give us the opportunity to place the Gaussian in this international workshop. So today we will discuss, as Dr. Hemant said, that some advanced topic in the Gaussian. First we will cover some introductory part of the Gaussian. I know you all are aware of what is the Gaussian, what is its capabilities, but still the new version has been released, or some minor revisions has been released recently in the Gaussian. So it would be very important for us to discuss on those topics. Then we will cover the onium calculations, right, for biomolecules. Then we will cover some excitation states methods like TDDFTs. Then we will discuss about the visualization of the orbitals, the interpretation of the energies, right? Then we will cover the reaction parts also, how we can predict the reaction, transition states, the IRC calculations, which is quite interesting part in the Gaussian itself, right? So before begin of the this session, I usually tell my audience that software is not a chemist, right? It's not a chemist. Software is basically a seeker, right, which is waiting for your instructions, right? Every software has their own capabilities, their own limitations, right? Gaussian is also having there's some limitations and the capabilities, right? So whatever the input you will prepare, right, don't expect that you are perfect in the Gaussian, right, or any other software, because it's basically your mind is perfect, right? Not your keyboard, right? Okay, so some citations. So Gaussian 16 revision C.01, right? So these are the contributors to make the Gaussian so popular, right? Then we have user resources, like Gaussian.com is the official website. Hello?
Yeah, so sir, your slides are not changing actually. Sorry to
not
interrupt.
changing.
Yeah, now it is.
I think it's taking time to.
It is working now. It is working. It is changing.
Yeah, now it is working. Yeah.
Okay.
Thank you.
Thank you. So Gaussian.com is what I said. Yeah, it's a quite popular website. So user manual, white papers. You should read the white papers in the Gaussian website. They have incorporated the some examples for the advanced researches, and also they have include some recent researches that are used by Gaussian. Some technical notes, latest product information, workshop announcement, and much more are there in the Gaussian website so that you can explore. Then we have Exploring Chemistry. So I don't know if you are aware, there is a book called Exploring Chemistry with theories like that, right? Written by Dr. Forsman. So this book is also very, very useful, and most of the examples I have prepared based on this book, as well as from Gaussian websites also, that I will show you in the demo session. Then, okay, Gaussian 16, you already know, more than 20 journals are there which uses the Gaussian 16, including the Journal of Chemical Information, Chemometrics, Computational Chemistry, Computer-Aided Chemistry, right? So Gaussian is basically the electronic structure program used by chemists, biochemists, physicists, and other scientists worldwide. Starting from the fundamental laws of the quantum mechanics, it predicts the energy, molecular structure, vibration frequency, and other IR, right? UV for that excitation energies. Then PBC is also there, periodic boundary conditions, that can also be predicted by the Gaussian itself. So what is it good for? So equilibrium, TSS structures, dipole, quadrupole moments, polarizability, right? So NMR spectra, electronic excitation, UV spectra, reaction rates, thermochemical data, frequency calculations. So all we can cover with the help of the Gaussian. Capabilities includes the determine most stable molecular geometric and energy, Raman, UV, NMR, optimizing the transition state, PCM, that is a solvation methods. So ICPCM, PCM method, SMD methods. So all these are the solvation methods which is there in the Gaussian itself. So ONIUM techniques, which we are going to discuss. Then we have a model surface using a 2D PBC periodic boundary conditions. Right? So then we have a Gauss view. Gauss view is basically the GUI, graphical user interface of the Gaussian. Right? The Gauss view, we can import or build the molecular structure that interested. Right? We can set up the job. We can visualize the spectra. We can visualize the orbitals. So all these visualization can be done with the help of Gauss view. The latest version of the Gauss view is Gauss view 6. Right. So this is basically the entire workflow. So we can import this structure, and then we can build or modify the structure. Then we can set up the job, then calculate, then examine result, and then visualization. This is our standard workflow to work in the Gauss view. So what's new in the Gaussian 16? TDDFT. TDDFT is also there in the Gaussian 09, but there are some extended features are there. Then we have enharmonic frequencies, mixed plot spectra visualization, PCM solvation cavity visualization, intermediate geometry at the certain time of 2 point during the optimization. If your jobs has been ended up, then you can start that particular job from their part also itself. Right? summarizing the result in the single window, like optimization result, thermochemistry result, and the converged result that can be seen in the single window. Some DFT functionals, M08 families or PW families are there. Saving movies file, .gif file, .mp4 file, PM7 solvation methods. There's no solvation method. PM7 semi-empirical method is there. GPU enabled K40, K80, P100 and B100 are compatible with the GPU or with the Gaussian now, right? Then we have symmetry point groups. So there are much more new features are there, so which we are not covering on this particular session. So now let me begin with the ONIUM calculation. So just a brief introduction of the ONIUM. What is the ONIUM, so that we can have some idea. So ONIUM is basically the hybrid method, right? The QMMM method, QM method, right? So like these hybrid method, the ONIUM is also a hybrid method. Suppose we have a single compound, right? And which consists of the 20 or 25 or let's say 40 atoms, right? We can do the DFT calculations. We can utilize the resources. We can utilize the memory, we can utilize the processors of the computer, right, for the DFT. And it may take like an hour, right, or 2 hours maximum to complete the calculation. But suppose if you have a protein molecules, if you have a, let's say, PDB file, right, having thousands or 5,000 molecules, suppose if you run the DFT calculation on that particular protein part, right, or PDB file, you cannot imagine how much time will it take, right? So that's why we incorporate the ONIUM Gaussian. So what ONIUM do is, so first thing is why ONIUM, right? Because the biological substances and the process includes commercial and research interest, right? Biological systems are obtained from X-ray diffraction or NMR techniques. We cannot deviate from this, right? Most biochemical reactions occur in solutions. Calculations on very large molecule may not be practical at the desired model chemistries. So the last point that we have discussed, DFT. We cannot utilize DFT to whole molecule. So what ONIUM do is it will part your particular file or your molecule or your system into various groups. There is a real system, there is a model system, and there is the intermediate system. Right? So there are some definitions like real system. What is real system? Right? Suppose we have a protein. Right? In protein we have some residues, some solvents, and the ligand. Right? So these all 3 or 4 parts comprise the real system or R. Right? But we are interested in more in the binding side, right, or the ligand side. So that is basically your model system. Right? So what we do in the ONIUM is we can do the calculation. That particular interesting part, we can do the calculation with the DFT or the Benedictine method, or for the rest of the part, we can do the semi-empirical method or MM part, molecular mechanics part. Right? So if you can see this diagram, the third layer, which is basically the environmental effect on the first layer, right? We can use the lower level method. So lower level, basically the QM or sorry, MM method. Second layer is basically if you are doing the two-level onion calculation, then it will be considered as a boundary. And if you go with the three-layer model, the electronic effect on the first layer is the medium layer. Then the first layer is basically in which the bond formation and breaking takes place, right? In this particular layer, we use the high-level method or the ab initio method, like DFT or HF particle. Right. And, here I have this is basically the simple example. Like it may be a protein also. So let's take an example of the hexane. So hexane, the 6 carbon atoms comprises the real systems, right? And we can identify some groups or in which we can add some other atoms and then we can predict the energy. That particular area of the hexane, we can consider it as a model system or high level, right? And then rest of the part we can consider it as a lower layer, right? So in the real system and the model system, we can partition, and by default, the Gaussian can attach a link atom or hydrogen, right? The partition basically. So partitioning defines the hybrid method and must remain constant in all species and reactions, right? So for the lower layer, we can provide the MM, and for the higher layer, we can provide the QM method. It continues. The next slide, if you can see the third carbon, right? You can see the H1, that is basically a linked atom that has been attached. Right? So this is the input example for the onium calculation. This is 2-layer onium. Right? So if you can see, there is a XYZ coordinates, right? And we have applied onium calculation, the MP2 and method. Now coming to the output file. So predicting the energy of the real system, predicting the energy of the model system, and predicting the low level real system energy, high level model system energy, and low level model system energy. So adding up all these system energies, we can get the onion extrapolated energy. So this extrapolated energy comprises the low real energy plus high model energy minus low model energy. Then there are some onium rules. Only use onium for relative energies and keep the partitioning the same for the various systems. These rules you can also find in the Gaussian website itself. Dr. Clemente has provided this particular rules in the book. So you can also go through the website itself. Then the next is identify the part of the system where the action takes place, obviously. Then in onium, basically in QM and MM calculations, the bond breaking and formation must take place at least 3 bonds away from the MM region.
So I just want to say something that yes, all the participants, wherever you are not understanding or you have any doubt, you are free to interact, because this is a hands-on session and you should understand whatever is getting explained. So you interrupt and then you can ask questions if you are not understanding anything. Thank you.
Yeah. Then okay, the rules basically the only cut non-polar carbon-carbon single bond, only cut the single bonds, right? do not cut the cyclic structure. So if you are, obviously it will disperse the charges if you are cutting the aromatic rings, right? I think I will show you the examples that I have prepared. Let's begin with the how to set up the onion calculation, right? So yeah, so let's say this is my butane, right? One butane. So if you can see, there is a wire structure and this is the ball structure, right? So the wire structure is basically the lower layer and the ball structure is basically the higher layer, right? I think it would be quite confusing, so I'm just making a new window and adding let's just profile, and then we can add the right, and then I can change the bond. Just go to the builder, modify bond. So between this and this, there should be a double bond, and then we can delete the atom. Right. So suppose this is my structure. Obviously, I can do the DFT, but this is just an example. Suppose this is my particular area which I'm interested in. I can attach some groups also and can predict the energies with the various group. So I just need to select this particular area, right? And in this particular area, I would like to set up a job with the QM method or DFT or HF method, and rest of this particular site, that is a W double bond, so I can skip it as a MM method, molecular mechanics method, or semi-empirical method, let's say. Right. So here in the Gaussian panel, control panel, you can see there is a select atoms by clicking, select atom by brush, select atom by clicking. Right. So let us select these particular area of the molecule, right? And now the next thing is go to the tool. In the toolbar, there is atom group editor. Right? In the atom group class, you can select the onium layer. Right? So by default, you can see it is red circles over each atoms. So by default, it is in the higher layer. Right? But we only need our selected area as a higher layer. Right? So just need to add selected atom to a group, atom group. Right? And then onium layer, the rest of the one. Okay. So this, and this. Okay, and just okay. I think there is some disturbance with the system, so okay. Oops, sorry. All right. So what I have done is, if you can see, if I just deselect all these things and lower layer. Yeah. Right. So select each particular area and then can set up the layers, right, by selecting the particular atoms, right. So for example, let's go back to the input file. So here what I have done is, in this particular area, this is basically the lower layer, and then we have a higher layer. And then when we go to the calculation setup, right, we'll do the optimized frequency in the method. So here in the method tab, there is a multi-layer ONIUM model, right, that you should check, right. So in a higher layer, we can do the DFT with the B3LYP631G basis set. And then in the lower layer, we can incorporate the semi-empirical method with the PM6. Right. Solvation, if you can add any solvation method, then you can also do here. Default is the IPSM method. Right. Then after setting up this particular job, we can go to the submit and we can save the file and the job will run. And then here we have the output file. Right. So if you go to the result, the summary, right. So yeah, this is single point energy calculation. And then if we go to the result part, the stream output result, right? So you all know that there is some philosophical quote, and make sure that normal termination would be there with the date and time. So the first thing is the citations, right? So this is all the citation. Then the input file, whatever the Z metric it will form. Yeah, so here if you can see, this is the link atom here, right in the lower layer for the carbon. Then we have a coordinates, distant matrix, right? Now we have alpha, virtual, occupied, beta, the special orbitals has been formed. So in the orbital analysis session, I will show you that this is the LUMO, this is the HOMO part, the occupied, virtually occupied, and the occupied one. And then going down, what we basically are looking for is the onium extrapolated energy. That, if you can remember, we are basically find the extrapolated energy of real system and the low system. Yeah. So here, if you can see, so grid method, low system model energy, right? Low system model energy. Model energy is basically the that particular area which we are interested in. Then method 2, high system model energy in the higher system. Then low system real energy, that is basically the all the system energy in the lower system. And then after adding all these three particular energies, we will get the onium extrapolated energy, right? So this is also the better way to reduce your time to calculate the whole system with the QM method, right? Then there is another application of the onium, that the vitamin E, right, tocopherol. So there are two conformers. One is having the oxygen in a upper ring or in a down ring, right? So after setting up the job with the onium calculation, so obviously we are more interested in the biphenyl ring of the tocopherol. So we will take that particular area as DFT or higher method, and rest of the tail part, right, we can skip it as a MM method, right? And then we can calculate the Gibbs free energy, and whichever the least in the energy, that will be a stable conformer. So upward conformer is more stable in the tocopherol stable. So if I can show you, this is the input file for upward conformers. So if you can see here, this particular oxygen is placed here, and this downward input file here. Okay. I think it's the same file. Okay, whatever. So let's check the onium upward, and the result is stream output result. Okay, I think it should be there. Optimized frequency. So here we have all the XYZ coordinates. From here, the optimization has been begun. Right. Oh, sorry. Yeah, so here you can see the extrapolated energy for this one. So that particular thermodynamic data you can find in a frequency calculations. So it will conversion in every convergence it will calculate the onium extrapolated energy. I cannot find right now. So what I would like to show you is that the thermochemical data with the Gibbs free energy, right? Whichever the lower the Gibbs energy, it will be more stable. This example you can find in the Exploring Chemistry book, right? That white color book. You can find this particular example that is very popular example, and it is also published in our Ocean website white paper, right? Similarly, we will talk about this PDB file, but before that, like this one, right? This is the alanine and the glycine moiety. Right? So if you can see, this is 3-layer model, right? So the phenylalanine part is basically it's basically the intermediate one, and this glycine part is basically the higher layer, and rest of the part is the intermediate part. So once we go to the calculation, so if you can see the high layer, medium layer, and the lower layer is there. So the lower layer we have incorporated the mechanism, calculation.
Hello, sir.
Then hello.
Hello, sir.
Hello.
Am I audible?
Yes, yes.
Sir, how you decide which is lower level, upper level, higher level, like?
Yeah, that is what. If you are more interested in the particular part, right, a section of the molecule which is more variated, right, more prone to attract the nucleophile or any, electrophile. So that particular area you can decide the higher layer, right? And rest of the part, for example, let's take an example of this tocopherol, right? So in this particular area, if you can see, this is the higher layer and rest of all is the lower layer. So why we done this higher layer is because this particular oxygen can be in the upper side here, and in the another conformer it would be in the in this side itself, right? So
in medicinal chemistry point of view, Monica, if your ligand is interacting with a protein, so the place where ligand is interacting, it will be at higher level and all the environment will be at lower level. So wherever you are having a strong attention, where you want more information, you keep it at higher level, and where all the other environment which is far from the interaction site or binding site, you keep it at lower level.
Yeah.
Okay. Sir?
So, yeah.
So this is Eileen speaking from IIT Guwahati. So,
yes.
Sir, where the intermediate level is required?
So intermediate level is basically partitioning level between the higher level and the lower level. So I don't think intermediate is basically required, but most of the calculation that I have
No, Eileen. So intermediate level, suppose you are having a ligand and you are having a protein, right? So ligand where it is binding, that area is higher level, and the immediate of that area, some environment where some kind of activity can be there from the ligand to the protein, some maybe 5 angstrom area from the binding site, it can be the intermediate level, and all the outer environment, it will be the lower level. So the main area, higher level, the immediate area after the main area, intermediate level, and all the rest of the environment, lower level. So that if any influence is there in the area, then it will be covered with the intermediate level and not with the lower level. So this is the basic idea behind this. This can be done only in Onium. So that is a very interesting part.
Yeah.
Okay. Thank you, sir.
So I think your query regarding the protein and the ligand. So as Sir said, that ligand is basically, we can copy it as a higher side, and if you can see rest of the protein part, we can do it as a lower side or lower layer, right? But before incorporating this particular refined structure of protein and ligand, what you can do is, once you open the PDB file in the Gaussian, there is an options, right? So once you go to the options in the PDB file section, here you can see the skip water molecules, right? You can skip the lone pair, right? Replace with the valence atom, right? Add hydrogen if not present. If yes, then you can select from here itself, right? Then click OK, then open your PDB file. And once open, you just check with the distance from the ligand to protein, right? As Sir said. Then you can set up the job, but this is not quite easy task in the Gaussian because it may take a quite longer time. Hit atom, it will be adding and then replacing the hit atom, and all these PDB modification would be included. So the ultimate purpose is to just go to the calculation part, then choose the method, then higher layer, which is in case our ligand, and the lower layer, which is in case of our environment and the rest of the proteins, right? And then doing this output file, we can find the result, and we can find the extrapol. So this extrapolated energy of all the real and the model system, right? So this is quite heavy file, so I think it has taken some of 1.5 hours to complete this particular reaction. It depends on you, means you can select that particular interesting area as the higher layer, and then you can use you can add the hydrogen or can skip the residue, and then you can go with the semi-empirical layer for the lower side, right? Okay, so this is basically the Onium that is ahead. I also would like to, I always do this. I always prefer to go with the Gaussian website because what are the recent advances there in the particular area. It will be there covered in the Gaussian website itself. So once you go to the I think it should be the sorry. '16. Indirectly, because that is quite more useful for you. Yeah. Right. So here we have a whole bunch of descriptions. What are particular keywords that is there? The onium, and then we have more examples over here, right? So here you can see this is 2-layer onium. So and then we have a 2-layer onium job, then freezing the atom, handling the SCF issues. And relay spin intensities, required input. So all this thing we can do, and then we have a technical note, investigation the reactivity in the spectra of large molecule with onium. Right? Because of the short of the time, I could not able to do this calculation, so that I can will share this one to you. Right? So this is how they have optimized with the reference structure, how much is the diffraction with the RMST calculations. So here if you can see the energy, how it can be predicted. Right? So all the DFT amber plus RT4 plus DFT in the real model system, amber minus RT4 the real system, sorry, in the model system. So this is how it can be predicted. Right. So here we have a linked atom, the linked host atom. It will be defaultly attached with the Gaussian. Right. Okay. Right. There are a bunch of tutorials over there. Right. I would like to request you to go with the Gaussian website. Try these examples. Make a job, and then you can calculate the energies, and then you can go with the reference part, and then you can just predict how much your result is accurate or how much result is matching with the reference part.
So one of our audience, they want to know how Gaussian is different than Jaguar.
I don't know the exact answer because I'm not a user of the Jaguar. But if you can just let me know what Jaguar can do in the onium, because.
So Bilal, Jaguar is given by Schrödinger and Gaussian is given by Gaussian, and both of them are having quantum mechanical calculations. Both of can do all these kind of calculations for what Gaussian do. It just depends on which platform you want to use. So if you want to run DFT calculation at BCLIP 611GD star level, there is no difference between Gaussian and Jaguar. This is for your answer. Okay, and then one more one more question is, what is the relation of softness of ligand and how to decide activity according to softness? I think this is not relevant to your presentation. So softness, softness can be calculated by using HOMO-LUMO values given by Gaussian. But then it's not always that a ligand can be correlated with softness. Sometimes it can be correlated and sometimes it cannot be correlated. And then one question is, let's say we need to determine and compare the energy level for two different protein complexes, protein drug, modified protein drug. How DFT could assist us? Can you answer this question, Charitra? So if there are two different protein complexes, one with protein and drug molecule and one with protein and modified protein and drug molecule, how DFT can assist? Bilal Ahmed. Bilal Ahmed asked this question.
Well, you can set up the jobs of all those two molecules, complexes, right? And I would like to suggest you go with the APFD method instead of B3LYP, because the APFD is basically the hybrid functions, which is basically used with the organometallics as well as the biochemical, like this one. And then once you go to the optimized frequency, right, and method, then B3LYP and the APFD method, right, this is the quite relevant to the biochemist, as I have seen most of the biochemists doing with the dockings and all. APFD and the CAM B3LYP, you can select these functions and then compare the energies and the the deviation in the energies, the optimization level of both the molecules.
And Nidhi is asking, not Nidhi, this is Akub Sarfaraj is asking, what is Hartree?
Hartree is the energy. So whatever like HOMO-LUMO and the the excitation state energies, so those all basically the units. Hartree is basically the units in the computational chemistry.
I hope she's not asking what is Hartree fog. She's asking only Hartree. So this question is not clear.
Hartree is basically the unit. The energy unit.
Yeah, fine. Okay, then please continue.
Okay. So next thing is basically what we cover is the TD-DFT, right? Okay. So
since we have a limited time, maybe you can see the real hands-on that how you prepare the input file and how to understand the output file and how to analyze this, and not the explanation part. So maybe input file and output file and analysis, so that we can directly understand how to analyze this.
Okay. So TD-DFT, basically the excitation state energy calculation, right? So DFT basically it gives our ground state energy, right? But for the TD-DFT, it will give the excitation energy, like CI method is there, Zindo method is there, TD-DFT method is there. So this is basically our main moiety or compound, then what we can do is, once we go to the calculation setup, no, I think it's not there. So yeah. Okay, so first I have just to optimize this structure, right? So that it can be go to the ground state, right? Ground state DFT with LYP I have done. And I think you must know the link 0 section, the memory limit, and you can also specify the processor, right? And if solvation is any, you can also add the solvation. I have just prepared in the Gaussian phase, right? And then we have ensured that our output would be in the ground state. Once you go to the summary, there should be imaginary frequency should be 0. If is it 1, then it would be a transition state. Then after optimizing this to the structure, the log file we have getting the log file, and then we can prepare that particular log file as a input file for the 3D DFT calculation. Then going with the energy calculation, right? Method, if you can see, Zindo CIS method, the TDSCF method we have chosen. Then DFT, the B3LYP 6311G, DP, polarized function, and then solvation, and you can also add. Then submit the this particular job, and then the output. The output going to the result, the UV visible spectra, right? Now this is the singlet, right? So here we have an ECD spectra also. The main thing I would like to show you is the output file for. Okay, let me just close all this file. So. Yeah. Okay. File and then result, stream output result. Okay. So I think we have taken a 6 excitation states, right? Once you go to the calculation setup method, yeah, solve for the excitation state for 6, right? And then result, stream output result, yeah. Now the stream output result, going to the, yeah, so here we have a occupied HOMO, virtual the LUMO, right? Then we have, yeah, XYZ dipoles, oscillation energy, right? Here you can see singlet, the energy, the lambda, right? The oscillation strength. So oscillation strength basically it gives you the it's a dimensionless function, and it will show you the how much radiation is absorbed or how much radiation is emitted, right? If it is near to the 0, we can skip those particular excitation state. We can, in this case, we can more interested in the excitation is 2, right? So it is going with the 39 to 41, right? And after getting all this part, let me show you the visualization, the CHK file. This is very quite interesting part, because now we are basically seeing that the transfer of the electron more towards this particular nitrogen area to this CN area, right? So once you go to the tools, the MOs, Right. Now in the MOs, so this is alpha MO. So basically, these are the energies, and these energies are in the heart rate, right? So you can see the here. Right? So the green partition is basically, this is the HOMO, this is the LUMO, right? Once you load the CHK file, and then okay, take time, and then we can visualize and update 13A and 40. Right? Okay, it's now loading. Yeah. So here you can see, let me just also update the HOMO +1, HOMO +2, LUMO -1, LUMO -2. Okay.
Hello, sir. I had a question.
Yes, sir.
Yeah, sir. I'm Pradeep from IIIT. So the question I has is with respect to TDFT calculation. So for finding the UV visible spectra, so how do you specify the number of excited states you should consider for the calculation? Like whether it should be 3 or it should be 6 or what number? How do you specify for UV visible? If I want to determine the UV visible spectra for a compound using TDFT calculation, how many excited states should I consider?
So default one is 6 basically, right? And it depends on the more chromophores you have on your molecules, right? So Professor Clemente and Forestman has comment in this that more the chromophores, more citation state you can incorporate, like 20 in the case of organic compound if you have, right? So it depends on you, means how much is the double bond occupancy is there in the compound.
Okay, thank you.
Yeah, and so if you can see here, the HOMO +2 and HOMO +1, then HOMO, and then LUMO. So you can see this part is going towards the cyno group. So this one, then this one. So let's take this one, right? LUMO. You can see the charge distributions from the nitrogen part towards the cyno group, right? You can also visualize in the cube part. So, for example, let me just do the surface contours. New cube. So what I can do is I will choose the total density, right? The SCF method, okay, and then with the CI method, right? Configuration interaction method and the SCF method both, and then we will subtract these two cubes in order to visualize the density distributions. Okay, it is now loading with the CF, right? And similar way, we can do with the I'm sorry, I think it's yeah, density. So for the density calculation, there is density is equals to current. If you know the population analysis. Yeah. So once we go to the summary, calculation setup. So if you can see the density current, right? So this should be there in order to visualize the charge distribution. So once we go to the result surface, the cube, new cube with the total density, CF, yeah, CFCI, okay. Okay. Just taking quite a long time. Okay. And then with the CI method, so like we are willing to visualize from ground state to the excited state charge distribution, right? And then we will subtract this SCF density with the minus CI density. Right, and then we need to load the new cube, subtract two cubes, electron density from SCF and CI. Right, and new surface, okay, and new matte surface with the excited cubes. Let me choose our difference between CF and CI. Okay, right. So here you can see the red part is basically the electronegative part. Right. Don't confuse with these. These are not the charges. Right. This is basically the you can say the numbers which gives you the negativity and the positivity, the sides, right? So you can see the charge distributions. The same manner you can see the ESP electrons. Okay, the ESP I will explain in the next example, right? In the similar way, what I would like to show you is the benzene state. Yeah. So we have optimized the benzene, we have set up the job for the TDDFT, and then we can have the UV visual. So if you can see here, there is a question mark, then SYM, symmetry. So symmetry is not known in this particular part, right, the UV. So what we can do in the next calculation, we can set up a job with the pop full keyword, right? So still there are some question marks are there, which cannot be predicted by the Gaussian, but the E1G symmetry is there, right? Like jarred and unjarred symmetry. Sometimes you can find the S1U like P1G like that. So pi orbital Girard, sigma orbital and Girard, like that.
Is it possible to show the picture in a little bigger size and difficult to read where you are, understand where you are clicking? So if you can make it this box bigger size, it will be very useful.
Okay. Okay, like this.
Yeah, a little bigger. So you can stretch it by clicking on the corner and you can increase the size. Yeah, fine.
So there's this one, and then what we can do is okay. So charge transfer and the TDDFT calculation. You can also do this similar way with the TDHF calculations, right? Zendo is there for the more excitation state for the semi-empirical methods. Okay. Well, when we talk about the Gaussian website, right? If I can show you some examples, more examples. The TD one, right? So the examples. Okay. So excitation energy, singlet A2. Okay, that lambda into oscillation strength, availability with these functions for the TFTs, the Hartree-Fock. Then we have options like solve for the M state, right? So all these are the energy ranges keywords, which then yeah, right. So the main thing is that first we need to just optimize the structure, taking back to the ground state, and then with the TDDFT calculation, make up with the energies, then select the TD-SCF method, then calculate one, get the UV spectra, and then if you would like to get the charge distributions, then set up your job with the density population, density current keyword, and then generate a cube. Then you can visualize the particular surfaces, right? Now, let me just go with the orbital one, because there is something ESP and this particular excitation is quite similar. So just for an example, I have prepared this file for the formal group, right? So if you can see, obviously oxygen is in the negative part, so for detecting the electrophile. So you also know this vibrations, you can see, right? The wagging and all this stretching, asymmetrical stretching, right? So that you can also get the spectra. So the spectra can be inverted to the axis also, right? So what I would like to show you is the surface and the contours cube. Oh, sorry, it's a log file. We need to go to the CHK file, right? And then we can go to the surface and the contours, new cubes. Let me just go to total density. And then visualize the total density, right? And then we can do map surface with the ESP, right? So here you can see the oxygen in the more negative part, and the bluish is the hydrogen part. Okay, that means so this way you can just visualize the more parts. Like if you can add to more electronegative atom, it will the surface will cover like the middle part of the red, right? So in this way you can visualize the things. Then what I can tell you is the molecular orbitals of the N2, right? So this is a famous example. So you go to the tool MO. So how many you can have now? If I just need to load the ones, and then I'll show you the s orbitals and the p orbitals. Let's update. Okay. Anyway, so here if you can see the s orbitals, right? So we have two nitrogens, right? So 1s2, 2s2, 2p6, like that. So 20 configurations, and then if we go above, we have this MO, right? So we have AOs, atomic orbitals, and then we have MOs, then we have pi bondings. So if you can see these four having the similar energies, so having this more dependent on the p orbitals, right? Px, py, pz orbitals. I think I can also write the Cartesian axis, right? Yeah. Then the LUMO is basically the unbounded forms, right? And if I also LUMO -1, right? Let's focus on the HOMO part. So it's basically the parallel to the pi bondings, right? So try with some small molecules, right? If you are a beginner, so like HF or like HCl or some other electronegative atoms, you can just try with those small atoms, and then you can visualize how the AOs, atomic orbitals, is forming the molecular orbitals, right? So this way you can see. You can also visualize these orbitals in NBO calculations. So it's natural bond orbitals, which shows the atomic orbitals. Okay, so first thing is the how we can set up the NBO calculations. So I'll just go with the optimization, the DFT, and then in the POP section, right, we have performed the NBO calculation, the full NBO, right, and then we have the tools, the MO, Right. Okay. Question load. It's okay. Right. So this is how we can choose. Right. So here if you can see the oxygen 1s, then nitrogen 1s, then we have CO double one 1s, then we have 1s this one, then we have pi bondings for oxygen, carbon oxygen double bonding, right? And if you would like to get the more depth in the calculation parts, then you can go with the stream output results, right? So this is the HOMO, this is the LUMO anti-bonding part, right? So because the stream output result with the log file. So here you can see the occupancy, the energy, this for delocalization, geminal, vicinal, remote. Lone pairs. You will not find this NBO in the Gaussian because that is basically the third-party software which is incorporated in the Gaussian. So NBO having the separate website, you can go to the NBO site and then you can get the information. This is Rabbit orbitals. Right. Now, okay, I would like to also cover the PST calculations. Right. So, because most of the chemists ask to how we can visualize the reactant and the product and the transition state within the same window. So I think you must all know this because that's quite easy for the Gaussian user, but still if I can show you the conversion of the CN2 isocyanide, right, the reaction. So HCN to CNH, which is quite a popular one. So what we can do is we can just begin with the with the carbon, and then so we have HC, and then we can add the nitrogen atom over here, right? Now we can also modify the angle first, so it would be 160 degree, right? Okay, oh, I'm sorry. So it's 160, right? Let me just visualize the labels and the symbols. So here we have SCN. Now what we can do is we can copy this to the control panel and then paste, and we can copy it, and then we can edit, go to the paste, add to molecule group. Right. So here, if you can see HCN, so this will be CH. So let me just do the builder, delete the atom, and we can just choose the nitrogen and this one. And then we can modify the angle to 160 because it should be same. Right. Now, if you can see H2, C1 and N3, C1, N2 and H3. Right. So this numbering is not matching in our reactant and the product part. Right. So what we can do is we should go to the there is an option called connection link or connection editor, right? So once we click on this, we can take a reference atom as 1 and enable the auto fixing, click OK. Right? Now if you can see the H3, C1, C1 and N2, N2, right? Now to begin with the calculation, what we can do is simply right click, calculation, setup, and then we can simply go with the optimization and the frequency, optimize to TSQS2, right? Because we have two specification for reactant and one is for the product, right? So cardinal nutrient synchronous method is there, so it may require the QST2 and QST3. So now when we go to the setup in the job, right? After setting up the job, we get the output like this, right? So if you can see, basically there is a migration of the hydrogen atom from the carbon to the nitrogen, right? So here we have a TS, right? So how we can confirm? We can go to the vibrations and there should be a negative frequency, right? So if you can see, it is how it is migrating from the carbon to nitrogen. Then furthermore, we can also get the
one question here.
Yes.
So how did you generate the transition state?
That is what I have done. So we have draw the reactant, right? The left part we have a reactant and the right part we have a product, right? But make sure the numbering should be matching. So we have.
Okay. So you did not draw the transition state.
Sorry.
You did not submit the transition state structure. Only the structure.
So, ma'am, actually there are two methods. One is QST2 and another one is QST3. So in the QST3 method, we require to draw the transition state guess, not in structure. It's basically the guess. So this is basically the QST2. We can also do this with the QST3. But how confident you are to draw the transition state? But this is my correct transition state. If you are confident enough, then you can draw the transition state guess, and then you can utilize the QST3 method, right? But to show you how we can predict the transition state, right? So we have a QST2.
This QST2 and QST3 calculation.
Yes.
If you, as explained by Charitra, if you know the transition state structure a little bit, you should run QST3 calculation. If you have no idea about transition state calculation, you know the reactant and product only, then you run QST2 calculations. The problem is that if the molecule is big enough, QST2 and QST3 calculations will not work. Most of the time they get fails. Like if you are using metal atom like ruthenium or manganese or many other bigger metal atoms, these QST2 and QST3 calculations will not be able to find a proper, especially QST2, it will say that you give an initial guess. And even if you give initial guess, it is very difficult for this. So for the smaller system, this will work perfectly. But for metal atoms and for a bigger systems, you need to draw the transition state calculations and run it for a proper transition state.
Okay, thank you.
Right. So now the next thing is to confirm the TS. So for confirming the TS, we will run the IRC calculation with the log file of the HCN. Right. So once we set up the job, the IRC calculation with the both direction from reactant to TS and then from product to TS, right? It will be reversible. So we have setting up the job with the intrinsic reaction coordinates, and after getting this IRC output, right? Let me just repeat with the endless, and then yeah. So here you can see how it is getting migrated from the carbon to nitrogen, right? And if I can also show you the IRC pathway, right? So this is how it is working, right?
Maybe you can see the input file of IRC calculation and explain a bit more about IRC. So IRC is to find the reactant, corresponding reactant and corresponding product from a particular transition state. Suppose you have a transition structure, you run IRC calculation in forward direction and in reverse direction. The structure you get in forward direction will be close to the product, and the structure you get in reverse direction will be close to the reactant. So sometimes you cannot be sure that this transition state connect to which reactant and which product. Then IRC calculation will be very helpful. You can run either like it is shown in this example, both the calculation together. You can run it forward direction in a separate calculation and reverse direction in a separate calculation also. Maybe if Charitra can show the input file, we can explain it a bit more, that how to run this calculation to our participants.
Yeah, so here if you can see the input file, the IRC. So if you can see the follow IRC both direction as Sir said, right? Force constant should be once, and compute more points, n is 10. So 10 points it will be covered, 10 n plus 1, right? 1 is the input file also. So if sometimes what happened that it will not reach your TS, transition state. So in that case, you can increase the point to 20 or 25 like that, right? And then you can restart your calculations, right? Okay, method, we are not going with the method one. So like that, we can set up the job. Another good example I can show you is the electronization. So we have, yeah, here, not here. So here, T2, yeah. So here, if you can see, this is the hydroxypropionate acid, right? And it is generating the furan ring as a product. And if you can see, the water molecule is separating out here, right? Here. So just be very careful that your labels and symbols are not deviating, right? So H12 is connecting with the O13, right? This bond is breaking, and then this bond between the O and H is forming, right? Right here you can see. So I have used the dihedral bonding. We have changed the dihedral bondings, and then we can draw this particular reactant and product, and then the same way we can go to the calculation setup, TS-QST2, right? It is automatically defaulted if we have a transition gas or QST3, right? And method we can set up. Then after getting this output file, we get this output file, right? Make sure the image frequency should be 1, the TS. So now same thing, we can get the IRC calculation, right, with the both direction, and if required, the more point can be added, right? Force constant should be once. Then we can do this IRC calculation output file. Now this is quite very interesting part. You can see how the water molecule is forming and deforming, right? I can also so at 0.0 intrinsic coordinate, this should be our TS, transition state, right? And this should be our reactant, and then we have product, right? Now you can see how it is separating out and forming the furon ring. Okay. Now you query regarding the QST3. So I have a simple example. We have made that carbene, right? So now this is the amyl group. Then this particular hydrogen is shifting towards this oxygen, and then this is my TS guess, right? So that I have also drawn. Make sure that symbols and labels should be enabled, right? So now you can see the transition state. So reactant, product, and TS. This should be the sequence, not reactant, TS, and product, right? So the sequence should be matched, and then similarly, once we set up the job, the QST3 is enabled, right? So we have the 3 specifications. So we can set up the job, right? With the particular methods. You can do this calculation with the DFT if your country is not so vast, or you can also do the OEM calculations. So now with the output file, so this is my TS. Let's go to the vibration frequency. Is the negative we have, right? So it is migrating like towards carbon, towards the oxygen, right? And then making this as a input file for the IRC, right? Both direction, right? And then IRC calculation is this is the output file for the IRC. Making it timeless, and then we can see this is the plot, IRC plot, right? At a particular point, it is optimizing and towards the TS calculations and towards the our product, right? So if you are confident enough, you can draw the structure of the TS. Then you can go with the calculations. Right? There are a lot of things that you can do with the Gaussian. So we have covered the onium orbitals, TD, QST, then NBOs, right? So there are a lot more things, but I think you are more lean towards the medicinal chemistry. So I think onium is quite very useful, provide the usefulness for you. So anything which I would like to cover, just let me know.
Sir, some questions are there. So Ranita Pal from IIT Kharagpur, she is not able to interact. She has written the question about this onium. She is writing this onium extrapolated energy will be equal to the energy if we calculate for the entire molecule without grouping into high and medium and lower level. So if you run a calculation without onium and if you run a calculation with onium, is there will be any energy difference? That's what her question is.
Yes, I think so, because the onium basically it is calculating the higher energy DFT calculation and the lower energy, and obviously if you are working with the DFT one, right, so I think it should be the different energy. Yeah. I can share some, I don't know if I can show you some examples right now, but
okay,
some benchmarking is there. So
Nidhi is also asking a question. She is saying that what these range of colors represent here in the mapped surface of the molecule. So there are mapped surface of the molecule. No, she want to understand the color coding, and also she want to understand what is POP-full. She is saying that there are two different for benzene itself. So can you please be clear about POP-full? How did you do that?
POP-full basically the density matrix. So the first question, I think this color coding with the surface map, right? So red color is more towards the electronegative part, right? So that it can be attract the electrophile, and the blue one is basically the positive part, which attracts the nucleophile, like that, right? But as I have told you, let me just to open that. Yeah. So once you go to the surfaces, cube, the density, okay, surface, right. So the color coding is basically default one. Oh, I'm sorry. Okay, new map surface ESP. Right, so this is what I think she is asking. So this red, do not confuse with these numbers, right? These numbers is basically not a charges. This is basically the system range that Gaussian has calculated, maximum towards the negative to higher towards the positive part, right? So if you can see, the color is basically going towards the blue one, right, more positive one, and the green is basically the intermediate one, and then towards the yellow and the red one. So obviously, if I can just visualize it as, let me just visualize the surface or mesh type, or I can do this transparent, right? So if you can see, this oxygen is basically more towards the, oh, I'm sorry, okay, right. So this is basically the oxygen, if any other electromagnetic part, like let's say if Cl or Br should be there, then also it will be the red part, because that is the electrophile, sorry, electronegative atom, and it is more prone to attract the E plus or electrophiles, right? So this is basically the color coding, but do not confuse that this is the charges, right? The charges, if I can show you, I think it's in the charge distributions. Yeah, this is the Mulliken charges, right? I can also, yeah, here we color with the atoms by charge, show the number, right? So if you can see the charge number, this is the charge number, this is not the charge, basically, right? This is not the charge, this is the charges, right? And regarding the pop full, so once you set up the job, right, uh, this population analysis, so full NBO, right, full NBO should be there, and level of output should be full NBO. So it will generate the population analysis, Millikan charges. Once you go to the output file, right? Oh, I'm sorry, this is the CHK file. So output file, I don't know if we should be there or not. Okay, I think I have done with the TD. Yeah. Yeah, TD full, yeah, this one now. So if you can see the population analysis, actually this is the energy calculation. So population analysis, basically it will form the density matrix, right? So per atom it will calculate the density, and then it will giving you the result as the population analysis, right? I cannot find any sample right now because that is basically in the energy section, so that's why.
So thank you so much, Dr. Charitra Gaur, for your this very nice hands-on on Gaussian. I hope it was useful for all the participants. This video is available on YouTube. You can always revisit that, and you can always ask question to Charitra wherever you feel that some explanation is required. So let us thank him. Thank you, Charitra, for all your time, and thank you so much.
Thank you. Thank you. Thank you.
Now we have 10 minutes break, and after that, we will be taking one session on Schrodinger, using Schrodinger for drug discovery. This will be taken by our group. So we will start Schrodinger session 5:30 p.m. sharp. It is 5:20. In 10 minutes we will start.
Okay, sir. थोड़ा शोर मत मचाना, ठीक है? पिक्चर देना ना ये। थैंक यू। रिकॉर्डिंग हो रही यूट्यूब पे आएगा।
Good evening, all. So, good evening, everyone. This is the last session for today's workshop, where we are going to explain about Schrodinger, how to use Schrodinger for docking and, if time permits, for virtual screening. Basically, we are going to explain the Maestro, the graphical user interface of Schrodinger program, which is available freely for academic use. Anybody can sign into Schrodinger and download Maestro. And Mr. Suyash Pant, who is from our group, he will be taking the hands-on session, and you are all welcome to interact during the session, and then, because this should be in a very interactive fashion, so you please stop in between, interact, ask your question, because if you wait till the end, maybe you will forget where you are supposed to ask the question. So interrupt in between and ask the question immediately whenever you have. Okay. So now, Suyash.
Yes, sir.
Yeah, please introduce yourself and then you can share the slide.
Yeah, good evening to all. I am Suyash Pant from I'm a PhD scholar there and working with under the guidance of Dr. Hemant Kumar Srivastav. So today I will be taking a session on how to use Schrodinger, how you can use the this free visualizer of Schrodinger to visualize your docking results and to make interactive graphs and all for your publications. Moreover, I will also going to discuss about how to use Glide, how to perform virtual screening, HTVS. There are 3 different modes in Glide: HTVS, SP and XP. Then how to rescoring using MMGBSA binding free energy calculation. And then what can be done, like if you have performed a simulation of 100 nanosecond and then again you want to calculate the binding free energy throughout the trajectory. What are the scripts available with Schrodinger? How to utilize those scripts? All these things I will try to cover. You can interrupt me in between wherever you are facing any kind of problem. So I will start now, sir.
Yeah, please share your screen.
Yeah, is my screen is visible?
Yes, it is visible.
Okay, so I will be taking on a hands-on on molecular docking using Glide, and I will be discussing
put it in the presentation mode, Suyash. Please put it in the presentation.
Sir, it is in a presentation mode only. It's not coming.
So wait, it will come in a few seconds.
Yeah. So meanwhile, I will discuss a bit about Schrodinger software. So it is commercially available tool for small molecule drug discovery. However, the visualizer is free for academics. So when I'm saying visualizer means what are the addition, what are the add-ons with that visualizer is like most of medicinal chemistry students, they are not into very well with the use of PyMol or VMD or Chimera and all. But I guess this Schrodinger visualizer is pretty much handy, and whatever you are keeping in your workspace can be directly imported to the PyMol. They have a very interactive session with the means you can add your visualization directly to the PyMol, and then you can generate the beautiful images for your publications. Sir, is it came to presentation mode or not?
Not yet. It is not in the presentation mode, so yes.
I think, sir. I don't know. It should come.
Yeah, now it is there.
But now I removed that from the presentation mode.
This is fine, so yes, please go ahead.
Okay, sir. So, yeah. So I will be discussing bit about the drug discovery. What earlier used to do and now what is the change in the scenario. Previously, like this cartoon represent like how Dr. Arnold Murray, means how he is conducting the experiment. He is randomly selecting a chemical from his shelf and then mixing it together and then checking whether is there any reaction between them, either he is getting any new molecule or not. But now the scenario has changed, and now drug discovery is like a very big term, and a lot of additions has been done. There are different branches, there are different domains, means every domain has their specific role into the in the pipeline of a drug discovery. Basically, computational method has been well discussed by Professor G.N. Sastry in the afternoon session, and where he has discussed about what are the ligand-based approaches, what are the structure-based approaches, what are the pitfalls and demerits or merits, what can be done, how to interpret the docking scores, and all these things. So I will be just briefly I will explain this, just what are the ligand-based approaches, what are the structure-based approaches. For structure-based approaches, the primary requirement, you need a 3D structure of your protein. So from where you will get the 3D structure of your protein? There are basically three different types of method. Either it is a X-ray structure, or it is an NMR structure, or it is a cryo-EM structure. If you have three different choices for a protein of interest where you have X-ray structure, NMR and cryo-EM, so it is suggested that better go with the X-ray structure instead of NMR or the cryo-EM structure. If you don't have any of these three structures, then then you can go for the homology modeling, but then you have to sacrifice with the quality of result.
Are not moving, so you are in the first slide step itself. Yeah, now it is moving.
I think it's not taking the presentation mode. So okay, I will take like I will present like this. So yeah, so there are three different methods where how you can determine the structure. These three are the experimental methods like X-ray, NMR and cryo-EM. If you don't have any of these three, means if nobody has solved the 3D structure of your protein, then you can go for the homology modeling. Then while you are using the homology model structure, then you have to look very carefully, because there are a lot of issues with the homology model. If your sequence identity is less than 50% or 40%, then you have to really take care a lot while modeling your protein. Apart from this, we have some handful of ligand-based approaches. First is the pharmacophore, means you can identify like what are the features available, what are the pharmacophoric features available in your ligand, and how they are arranged in the 3D space, like hydrogen bond acceptor, donor, hydrophobic, or ring aromatic. Then there comes a method QSAR. So here, if you have a set of series, congenering series, and all and the bioactivity is being identified based on a single assay method, then you can go for the generation of QSAR method. It is a kind of regression method where you can predict the bioactivity of your unknown molecule by calculating the physicochemical parameters of your molecule and correlating that with the biological activity. Then comes the similarity search. As Dr. Sastry has also stated that similar kind of molecule binds in a similar fashion, tends to have similar kind of activity. So you can also look for the similar kind of molecule and then predict what target or where they could bind. And then you can compare with the standard molecule or the crystallized molecule. If you have any crystal structure data, then you can compare your molecule, and there are a lot of studies has been already published in very good journals where they have identified the target for their molecule based on the similarity search. So basically today I will be talking much about the molecular docking. So molecular docking, previously it was known as a lock and key method, but then somebody told that while ligand is binding to the protein tends to change its conformation and make it fit well inside the binding site, and then it is called as the induced fit method. So this is one of the in structure-based approaches. One of the demerit is we cannot go for the induced fit approach for a large library of molecules, as it takes a lot of time. So basically, usually we consider our protein as a rigid and our ligand as flexible. However, you can always go for the induced fit method, but only a few number of ligands can be done. Means induced fit docking is computationally very costly, computationally very challenging. So mostly in a drug discovery, we use an integrative approach where we utilize ligand-based method, where we utilize structure-based method. But you also have to look into like which method to use at in which stage. So all those things I will try to discuss with you today. So going ahead, so this is like from how you can get the free access to the Schrodinger visualizer. So already actually I have shared the Windows version. So for Linux version you can download. You can go to the Schrodinger site, go there and create your user ID and password, then directly go to the free Maestro and then you can download. For 1 month trial, you can write to Miss Selvia Malik and write to her, and probably she will arrange a 1 month trial license for you. So this is the basic interface of Maestro after you opened it. So I will just show you the basic interface right now, and then we will go to the practical session. So there are like if you go to the file, it is simple like how you are using the Microsoft Office or Microsoft Excel, something similar like that. So then in Windows you have to change the directory whenever you are working, otherwise everything will go into the documents into a particular directory name as Schrodinger. And if you are working in a multiple project, then it will be a mess. You will not recognize what file is for what project and what file for what project. So this is the interface where this white portion is the workspace and this portion is your project table. So whenever I am saying that I have selected the things is the project table, it will be selected here, and if I have saying that it is in the workspace, it will be here. So because it will be very useful while you are working on Schrodinger, while you are working on docking or any of the module if you are using. So these two things are very useful, this project table and workspace. So for basic docking experiment, you need, first thing what you need is your protein. Second thing is you need your ligand library. Both need to be prepared because the crystallized protein or the experimentally identified by NMR or cryo does not have all the information. Means you cannot directly go for any kind of experiment. You need to prepare that. So when I am saying prepare that, then there are some steps are there, 2-3 steps are there in Schrodinger. Like they have integrated everything into a one module, that is protein preparation wizard. So there are 3 steps. First is import and process, and then you will fix some of the issues, like if there is any disulfide bond, if there is a metal atom, or if they have mutated the methionine to selenomethionine, or if side chains are missing, loops are missing, terminals you have to cap. So all these things I will show you one by one in the practical session. Then in review and modify, in this part you will get the chain names, how many chains are there, either A, B, C or D. Then here you will get the how many water molecules are present in your protein, and then here you will get the information about the heteroatom. So in the first step, delete water beyond, means if there is a ligand inside your protein, it will keep only those water which are nearby 5 angstrom and delete rest of the water. Why we are keeping beyond the 5 means near 5 angstrom of the ligand? Because water molecule sometimes form hydrogen bond, and water molecule is very important for like binding of ligand to the protein. Then in the last option, in the refine tab, we will refine the position of hydrogen. As hydrogens, you will not get the electron density maps of hydrogen in structure. So we will optimize, we will refine the position of hydrogen which we have added, and at the last we will minimize our protein, whatever we have make the fix, like if there is any loops are missing, any chains are missing, we have added all those things. At the last, we have to minimize that part. Then the second option is you need to generate the grid. So there could be two different options. One option is there is ligand already available with your crystal structure. Then it is pretty simple. You just select your ligand and it will generate a grid. Second option is you don't have the ligand. Then you have identified the binding site. So there are handful of tools like sitemap, docsite. So all these tools are there where you have identified the binding site. You have searched for the binding site and it gave you some certain output. So there you can just select that binding site as an entry. Like here it is showing molecule. So another option is there is an entry option. So you select that entry and it will create the binding site considering that entry as your site point. So now now you don't means you don't want to go for the sitemap, but you know that what are the coordinates or what are the residues. Like somebody has mutated, somebody has done some mutational study, and you know that these are the residues which are very much important for the binding of a ligand. If we mutate this particular residue to alanine, there is a loss in activity, or there is a complete loss of activity, or reduction in the activity. Okay, so there are three options. Centroid of the workspace, like with the first option what I told, you have the ligand inside your protein. So this is the default option. Second option, centroid of the selected residue. You will select those specified residue and it will generate a box considering all those residues as a grid point. Then in the third option, you know the XYZ coordinate, means not the XYZ coordinate, but you know a particular atom. Okay, so you will select that particular atom and it will create the XYZ coordinate. Then you can adjust the size of your box. Okay, then there are another options are then there are three other options: constraints, rotatable groups, and excluded volume. Mostly constraints, when you know that this particular residue is making a hydrogen bond or this particular metal ion is making any kind of interaction with your ligand, and it is important. If your ligand, and if you have millions of library, and you need to focus your search within a particular region, then you can just keep that particular constraint. Like if that particular interaction is there, keep that pose, otherwise discard that pose. Then rotatable groups are like what are the groups nearby your binding site, and if you want to add other rotatable groups, you can add it. Excluded volume is your binding site is very big, and you know very well that this particular part ligand should not go. So you can keep some spheres there. These are just spheres, dummy spheres, so that they will engage that particular part of the binding site and your search area will get reduced. Third option, you have generated the grid, you have prepared the protein, you have prepared your ligand library, then you can directly go. Then there is another step is, last step is the glide docking. So glide, three different modes of docking. One is HTVS, high throughput virtual screening. Second is standard precision, and third is XP, extra precision. HTVS and SP are very fast. Like HTVS takes around 1 or 2 seconds for a single molecule. Standard precision takes around 5 to 9 seconds. XP takes around 90 seconds for one single molecule. So it's like whenever you are increasing the accuracy, then you have to means that it will take more time. You have to compromise with the time, you have to compromise with the speed. So it is always suggested, first go for the HTS, reduce the size of your library, go for the SP docking, then again reduce the size, then at last you can go for the XP docking. After this, you can go for the rescoring of your molecules using MMTT delta G bind or any other method. Okay. Now we will use the practical session on how to use glide. Just a minute. Okay. So this is like I have already imported, so I will close this. and I will tell you how we can do it. So to open the Schrodinger, we have to go to the your terminal. So this is my installation directory of Schrodinger, and I will directly open this from here. Uh, Hello, is my screens are not visible?
Suyash, can you hear me? Anvesh,
Yes,
it's Anvesh.
sir. Yeah.
You do one thing, you cross this, you close this screen, then only it will be shared. It's not like Meet, it's a different platform. You close this first of all. Yeah, close this.
Okay.
Yeah, right hand upper corner.
Okay.
Close this entire and reshare the current that terminal thing. Then it will be visible.
Okay.
Yeah.
Okay. Wait.
Okay. All the best.
Thank you, sir. Is it visible now?
Yes, it's visual. I can see your desktop.
Okay. Yeah. So I will move to. Is this black color is visible, or I need to change it to white background?
We can still see your desktop, Suyash. The terminal is not yet visible. You go to the three dots, this share. Go to the three dots, and it will be displayed with different terminals opened. You select your terminal from that, not the desktop.
Okay.
You go to this open share tray near the mic icon in the front.
1 second, sir.
Okay, take your time.
Is it visible now?
Yeah.
Hello.
Yeah, we can see the protein and glide interface. Yes.
Yeah, I think it is giving some problem with the Linux interface. Right.
Okay, fine, no problem. All the best.
Yeah, thank you. So, what I was talking, okay, yeah. So I have downloaded one protein. This is the human MDM2. So this is the X-ray crystal structure. I prepared everything. So I will just show you the options which I have used. So there are options like to download here, you can give your PDB code, and then there are some options like diffraction data or biological unit. This data is like if you have the X-ray crystallographic data and means there are some missing loops or residues and you need to modify that using Primex, then you can download that data. Otherwise, if you are interested more into the docking and all, you can directly just skip this part and just import your protein. To import that, you need a working internet connection because it will connect with the RCSB database and it will download the protein from there. Then there are add hydrogens, as we don't have the coordinates of hydrogen atoms in the X-ray crystallographic data or NMR or this cryo-EM. So you have to add those hydrogens. Then if metals are there, then you can click on this particular option. Any disulfide bond like there are for in case of G protein coupled receptor, you know that this particular residues, if means here you will find one disulfide bond, if or there is a possibility like 2 cysteine are very near, so they will tend to form a disulfide bond. So for this you need to select this particular option. If it will find that particular distance, it will create a disulfide bond. Later on you can check in the literature or in the experimental data whether there is a disulfide bond is there or not. Then sometimes people use to mutate the methionine with silyllomethionine to speed up the crystallization process. So you need to convert those silyllomethionine to methionine. Then fill in the missing side chain and missing loops using prime. So mostly you will not get the that is a highly mobile region of your protein. So most of the time data. 20 residues, then it will not resolve that particular row. Then you need to again go to the homology modeling. You have to important. Cap terminis are it caps the N terminal and C terminal of the protein. Not get react during any kind of calculation. But if N terminal or C terminal is a part of binding site, then you need to think actually, because sometimes those N terminal or C terminal they tends to make some hydrogen bond with your ligand. Then you need to no need to cap that particular part, because then you will be getting a false data. You will get that particular interaction and it will get you will visualize that particular part in your docking score or any kind of calculation you are seeing. If you are focusing mainly into the binding of your ligand with the protein, that water is of no use. So you can just simply remove that water, remove those water. Then heterostates are for like that I will discuss in the depth. Then in the review and modify, if there sometime protein gets crystallized in multimeric form, so you need to chop it down into a single unit until or unless you know that dimer is means the ligand is binding between the dimer interface of a protein. If it is of no use, so then you can delete those particular parts. Then here you will get the information of ligand molecules. how many ligand molecules or SO4. It took all these things as a ligand molecule. So once you will select that, it will show you whether how it looks like, what is that ligand molecule. So okay, yeah, these are the water nearby your ligand. These are all the water present in your protein. So I have prepared that, and this is the refined picture of our protein with ligand. I have deleted all the water molecules, so it was not needed here. I was not interested in understanding how water molecules are interacting with the ligand, but whenever there are water molecules in your binding site, you are visualizing any kind of interaction, any kind of bridge between your by those water molecules, then it's better to consider those water molecules. You can perform the calculation in presence or in absence of water molecule, and then you can categorize it like what are the changes in the docking score when you consider the water, what are the change in the docking score when you deleted those water, and you can explain that later. Then in the refine tab, there are sample water orientation, use crystal symmetry, minimize hydrogen. So basically these options are to refine the position of hydrogen atom. Prop K is to identify the protonation states, to identify the pKa states of particular residue at at pH. Mostly people used to take pH at 7.4 or 7.0 until or unless it is mentioned in the crystallographic experiment that we have crystallized this particular protein as 6.8. There are several examples where there is some particular in case of proteases where they are catalyzing the reaction, then you have to consider that particular pH where that particular histidine or that particular residue gets activated. Then there are options like how to minimize. After making all these alterations, then you need to minimize your so after minimization we got something like this. And these are the this is the panel at the last in the right corner for the interaction. There are some non-covalent interaction, pi-cation interaction, and contacts and clashes. So 3 different categories are there. Hydrogen bond, salt bridge. If you want to change the color, you can directly click on any of the interaction, then you can change the color. This is to change the width of the this interaction line between your residue and your ligand. You can change its width by changing this particular line. You can manage it according to your need, how you want to, like in your picture or in your snapshot. Then this is a 3D visualization of ligand and protein complex. If you want to visualize the protein ligand complex in 2D, then you can click on this 2D workspace. Yeah, and here you will get the interaction in 2D. You can click on this LID, so it will show you like what are the residues, whether it is hydrophobic, whether it is polar, charged or charge negative, and by clicking on file you can just simply save your screenshot and you can keep that in your presentation or wherever you want that. To click the 3D representation, you can go to workspace and you can just simply save. And as I suggested that you can directly import your workspace visualization to PyMol. Here is send to PyMol. You have to specify the path of your PyMol directory and then you can just simply launch and it will open that there. I will not do that here now. Then comes to the glide grid in the second option. So here you can go to the glide and here is the option for receptor grid generation. So as I told, there are 2 different options, molecule or entry. If I have submitted the sitemap calculation, it will give me different entries with different scores for the binding site volume, binding site score, D score. So then I can select that particular entry. Here I have selected, I have the molecule in the binding site, I can select this. Now it is taking this is a very big box, so by default it takes at 20 angstrom. If you want, you can change this. You can change the size of this particular box. Then there are two other options like centroid of selected residues. Here you can directly select the residues which are interacting. Like in this case, I found that this particular residue is, sorry, yeah, pick a residue if you want to pick from the workspace. If you know the number of that particular residue, you can just give 45,67 and add it, and it will select. Yeah, and just OK, and it has selected, made a box of around 20 angstrom. So you can change that according to your ease, and you can simply delete it. So I will go with the centroids of the workspace ligand. Here you have to give the name of your job, and Schrodinger doesn't take the space, so you need to, every time you need to give, like if I want to give it like 4LW. Then there are three other options like constraint. So positional constraint, hydrogen bond and metal coordinate. So we don't have any kind of metal complex here. However, hydrogen bond constraints I can give with this particular residue. I think this is histidine, and these are the rotatable group. Except this, if I want any other group to consider as a rotatable group, I can just click on this pick group and I will select here in the project table, and it will be highlighted here. Excluded volume, as I told, like if you know that this particular part of the binding site is not important, because grid generation, it generates a rectangle box. So there is a possibility that that part of not your interest will come inside the binding site. So you need, so whenever your docking algorithm will work, it will search that particular part also as a binding site. It will consider that particular part as a binding site, and it will search the conformation of that particular ligand at that particular part. So it will directly proportional to the computational time. Means the big the binding site, it will also increase the computational search for that particular part of the binding site. So you can put the constraints at that particular binding site. You will tell your algorithm that you don't have to search there, except that particular part, you search the complete area, complete binding site. So means I have told that means we have performed some similarity search based on this particular ligand, which we named as a reference ligand. So it looks something like this. It is a spirocyclic molecule. Then we select, we took the Campbell database and we go for the similarity search for this particular molecule. And based on the similarity matrix, here is like the top molecule is around 78% similar with the this reference molecule, and at last it is like 10% similarity. So we prepared all these ligand molecules. We prepared all these ligand molecules using LickPrep. So LickPrep is also very simple. There is no much things are there. So as I told that you need to understand what is your project table, what is your workspace. So in workspace, I have selected only single ligand, but in my project table, I have selected around 2000 ligand. So this is what you have to give, or directly give the file where your ligand molecules are there. Like I can give particular for my for the preparation. And
hello, sir.
Then yeah, hi, hello.
Sir, we are unable to see the panel, sir. What you are doing, the procedures.
I have to give each and every time. Now visible, only panel is visible, right?
Yes, sir. These panels from the first onwards we are unable to see, sir.
So only the panel is coming now.
Yes, sir.
Okay, so this is the panel for the leak prep. So as I told, there will be three options: workspace, project table, and file. So project table, I have selected 2000 molecules. Can I share this? No. Okay, so this is the leak prep interface. So there are very basic options like if you want to generate the for your molecule at particular pH, whatever pH you can give there, it will generate states based on that. If you want to neutralize your complete chemical library, you can utilize that. And if you have already made your molecules based on like whatever charge should be there, so you can just simply do not change anything. Whatever you are feeding, just minimize that particular conformation or just minimize that particular structure. Then there are three options: retain specific chiralities, determine chiralities from 3D structure, generate all combinations. So retain specific chirality, what it will do, if your molecule is having one or two chiral center, it will consider only those two chiral center, means one single chiral center, then another chiral center, and generate multiple permutation and combination based on those two chiral centers. In the second step, it will just take the chiralities from 3D structure. Means first option is for whenever you are giving only 2D structures, not the 3D structures. Second option is when you are giving the 3D structure, it will only minimize that 3D structure. It will not generate different conformers for that particular molecule. It will just minimize that structure. And the third one, it will generate whatever combinations are possible based on 2, 3, how many chiral centers. So it will generate all the combinations. You can limit the combinations by changing this number. Default is 32. Then output format, you want it in Maestro, you want it in SDF. So it depends on you. Maestro will be much less space consuming because it is the GIF file, compressed file, and SDF will be more memory file. So this is the basic, and here you can change, like default it will take only single processor, means it will split your job into one single job. You can change, like in this system I have 12 processors, so I can say out of 12 you use only 4 processors and split the job into 4, like each job will take only single processor. Okay, so you have to manage accordingly. If you want to finish your job bit early, if you have like 1000 molecules and you want to prepare those molecules, wait, I think this, I will share this. It's not coming. Can you see the this means? Hello. Hello.
Yes, we can see this leak prep slide correctly.
Other than leak prep, you cannot see anything, right?
No. At a time, only one slide will be displayed.
Only one slide can be displayed. Yeah. So actually, it has it has multiple options, multiple modules actually. So every time I have to.
Yeah. Every time you click and a window opens, you have to select that window and that will be displayed.
Okay.
Yeah.
Okay, so it is not taking like from how I can do the how I can change the processor number. So you can click here and it will show you the job setting. Can you see this job setting tab here?
Yeah, it's visible.
Okay, yeah, so you can go to the job setting and then you can split your jobs like you want to. If you have a large library like 10,000 or 20,000 molecules, then you need to split your job into smaller jobs. So you can split that. Whether you have 4 processors, then split your job into 2. Just spare 2 processors for running of your interface and everything. If you have 12, 20, 30 or 40 processors, you can split your job accordingly. So after leak prep, you can go to the glide and ligand docking. So I will share the interface. Just a minute. Yeah, so it has multiple options. Like first option is you have to give your path for your grid file. So that you can take from here, or either you can just simply browse. Okay, so I cannot show you because again I need to switch. So I will just share with you. So then you have to give the path of your grid. Grid file will be your zip file, and whatever job name you have given, it will be inside that particular directory. Then you have to select the ligand files. So there could be 4 options. One is workspace entry, then second is project table, third is files, means directly after leak prep you can go to that particular directory and there will be one file with that out.megz extension. So out.megz extension means it is your output file. Then fourth option is phase database. Sometimes people used to generate phase database, means if you have a use library, in-house library, you can just feed your molecules and make a phase database. So every time it will fetch that phase database, and then you can go for the multiple, means right now we are selecting manually single file, 10,000 molecules from project table, and then 1 or 2 molecules from workspace. Phase database is like a remote library in your institute or somewhere else. It will fetch that database from there and it will give you the result. Then there are certain options, like as I told, there are 3 different modes: HTVS, SP and XP. Then fourth is the S-peptide, not useful for now. So HTVS and SP are similar, the scoring functions are similarly same. For XP, it takes much time, higher penalties are there. So all those things. Then in the output panel, you have you have to select whether you want pose viewer file, like where only the receptor file will be there, one receptor file, and then the dock poses of ligand, means you will get the coordinates and then you can superimpose each ligand into your protein. And in the second option, you will get only the ligand, means it will not consider the receptor, only the ligand coordinates you will get in the output form, and then you can superimpose those particular ligands in your initially minimized structure which you have used for grid generation. Then number of poses, if you want to limit, like you have 1 million molecules, and after that, means you want to limit that after output you want only 10,000 molecules or only 5,000 molecules. Then for how many poses per ligand you want, whether you want 1, 10, 20, means it depends on, like if you have 50 molecules, you can generate 50 poses. Okay, but if you have 50,000 molecules, you will consider only single pose, because 50 molecule, it will exponentially it will increase. 50 into 50, it will generate n number of poses actually. So you should have that much memory also in your system. And if for one molecule it is generating 50 poses, it will take that much time also. Then whenever you are going for XP docking, you can means apply strain correction terms, then write per residue interaction score. Like if you have you know that these particular 4 residues are very important for the binding, so you can consider those particular residue, and then you can compute the interaction scores, whether your molecule are interacting that particular. So these all things are helpful when you have a large library and you want to chop down for your further next step. So you can utilize all these options. In the XP docking, you can compute the descriptors actually. So this option will open whenever we will go for the XP docking. So what these descriptors will calculate means it will be helpful for the post docking analysis. So post docking analysis, it will show you means whether your ligand is going to the solvent accessible region. So if your ligand is going into the solvent accessible region, it is a penalty for your ligand. It will directly correlate with your docking score. Then is there means how many what is the score, what is the interaction energy with the means hydrogen, what is the interaction score of your hydrogen bond, then salt rich. So all these descriptors you can calculate from here. So I have performed HTVS docking for around 1721 molecules. So I will just go. So all these are the similar molecule which I have shown you. So I will be showing you first the scores. So,
Suyesh?
Yes, sir.
So if you share your screen instead of your presentation, whatever you will do, it will be visible. If you share your desktop.
In only every time it is asking for another thing actually.
No. If you stop the presentation and share your screen, the desktop.
Okay, now it is visible. Yeah, I think now it is visible. Okay.
Yeah. Okay.
Yeah. So we have performed the docking of so around 721 molecules. So in one by one, if I will analyze the poses, it will take a lot of time. So if you see, it gives you 3 different scores: glide score, docking score, glide G score, and glide E model score. So glide score and glide G score are same only, docking score. Glide E model is like for one molecule, it has generated 5 different poses. So glide E model is scoring those poses. Like for one molecule, I have 5 poses with like -75 glide E model score, -72, -73, -75. So it will score, it will take the best glide E model scored pose, like which is more in the negative. It will take that particular pose, then compute the docking score. And docking score is for to further score the different molecules. So did you get this particular part? I think it is a bit confusing. So if anybody has confusion in this, you can interrupt me now. Hello. Okay. This is a very small data set, so I directly go for the SP docking of all these molecules. After docking, I dock the reference ligand. Also, in this case is the co-crystallized ligand. So to superimpose the pose, means mostly all these ligands are very similar. Hello.
Yeah, we can hear you, Suresh. We can hear you.
Okay. Similar molecule, 62% similar molecule. If I will superimpose a less similar molecule like 39%. So if you see, the docking score is -2. Okay, so I will just change this and my protein to, okay. So these two poses are very different, and if you see these two molecules, So these two molecules are very similar, but still their dock poses are very different. Yeah. So I forget few more things actually. I will just tell those also. So lost means I just so there are quick selections are there. P for protein and style tab. And then there are different visualization options for ligand, wire, or if you want like this, and then when CPK. So if you want to hide all the hydrogens, you can hide those. If you want to show all the hydrogens, you can show all the hydrogens. If you want only polar hydrogens to be visualized and delete rest and just it will change the color scheme from carbon to green and light green for fluorine, and this will be for chlorine. Yeah. So these all are, so now if I want to select only the residues which are near so you have to select your ligand, then right click here and extend the selection to 5 angstrom. It depends on you. You want 3, 4, 5 or 6. Basically, default means basically you should go for 5 angstrom. Most of your interaction will be covered in the 5 angstrom area. So you can select that and then you can click on this I and it will give you all the interaction, all the residues which are nearby 5 angstrom. Now after that you can click on the P again, then hide all the hydrogen, so it will clean up the visualization a bit, and then you can select, like, you can change the visualization of the residues to thin tube and click anywhere in the workspace. So now there are a lot of unnecessary residues are also particular part, so it will not give me a very good result. So what can be done? Just a minute. Yeah, so what we can do is and then
Sir, your voice is breaking a lot. We are unable to follow.
लाइट से टक होती है.
Sorry for the technical glitch. And so yeah, what I was saying, you have to press the control and then you select only there is means whatever you have selected, it will deselect that and except select rest of the part, and then you can go to the style, click on the side and just remove that, and then you can click into the ribbon, click here, hide all the hydrogens. Now if I want to change the color of my protein means the ribbon. You click on P, go here, go to the ribbon, make it single color, and whatever color type you can make it. And now it is little bit
Hello, sir. Hello, sir.
Suyash, I think you are disconnected or what? सुयश का मैसेज आया है. So I think Suyash is gone, and whatever he has explained till now, it is available on YouTube, and you can practice it by downloading free Maestro. And if you have any question or if you have any concern, you can always write us. And we hold for 5 minutes, and then we will announce for the end of today's talk. Tomorrow morning, we have a very nice talk from Professor GPS Raghava, who is a renowned bioinformatician. He has developed more than 200 web servers, and his citations are over 15,000, and he is one of the pioneers. Please join 9:30 a.m. sharp for his talk. This will be followed by talk of Dr. Deva Priyakumar from IIIT Hyderabad, and then Professor Hajmi Hirao, and Madhvi Shastri will give a talk, and then it will be one more talk from Schrodinger. So that is not talk, that will be hands-on. So let's wait for 5 minutes and then we can close.